A very big hello to my wonderful champions out there. So, okay. Happy Makar Sankranti as well as Pongal to everyone who is watching this video. And Tolly ma'am, please provide practical syllabus for KCET 2026. So my dear and Oli, the information brochure is yet to arrive, and after that I will be definitely providing you with the practical syllabus. I am 02, a very big hello dear. Chandra Kant is also here. Good afternoon dear. Harshvardhan is also here. Just a second. Yes. Every one of you who are here with me, thank you so much for being there. And yes. No ma'am, you can take it. Good afternoon, Harshvardhan. Hi. So children, my dearest little children, make sure you share this video's link with everyone out there, your friend group, and then we can begin up with this particular session. It's going to be super amazing, super fun, and yes, super interesting. Yes? Subhash is saying happy Makar Sankranti. A very happy Makar Sankranti to you too. Lavanya is there. Zohir is there. Hello. So my dear children, for today's chapter, I have taken it. Happy Makar Sankranti. Oh my God. Yes. So today's chapter that I've taken is Principles of Inheritance and Variation. You know it's super important, right? So Principles of Inheritance and Variation in PU2 contains 13 marks. And by this video, I am going to make sure that you get these 13 marks. There will be one 1-marker, one 2-marker question, and two 5-marker questions coming off from this chapter, and we have to grab all of them. If at all you talk about KCET aspect, in KCET aspect also this chapter is super important. Why? Because over the years, the number has been just increasing. So it was 4, then 5, 4, 4, 4, and then again 5 questions have come from this chapter in the previous year. So we can expect the same. This is one of the most high weighted chapter in your KCET as well as your PU2 board. Okay. So I hope that is done. So shall we go ahead, everyone who is watching this video, shall we go ahead? So I'll tell you the flow of this particular live. So first thing that I'll be showing you is a concept. Okay, I'll say that concept 1 is what? 7 pair of contrasting character. Then basically I'll be explaining that a bit, and then we will start solving the question. So concept, question, that is how it is going to go. Clear? Done? If at all it is clear, can you give me a thumbs up? If this thing is clear, can you give me a quick little thumbs up? 17 people up there. Yes, that's the wiggle. Okay, let's begin then. Starting with the first concept that we are having, and that is 7 pair of contrasting character. You know, Gregor Mendel was planning and was working on a garden piece, which is also known as what? Pisum sativum. Pisum sativum. He wanted to prove something, and for proving that, obviously he had to do some experiments. And you know what? This person was such a hard worker. He basically put 7 long years on working on it. Yes, I'm not joking. From 1856 to 1863, he kept on working on this particular plant, that is Pisum sativum, and that was not at all a small deal. Why not a small deal? Because can you spend 7 years for on something? Obviously, no, it's too difficult for us, right? But he did. And when he did, he chose Pisum sativum plant because it had 7 pairs of contrasting characters. As in, for example, if at all I say height is a character. Okay, height is a character. In height, there are people who are tall. In height, there are people who are dwarf. So height is what? A character. What about tallness and dwarfness? They are going to be the traits. They are considered to be the traits of this particular character. So we had 2 traits out there for each of the character. And what were the characters? It was flower color. In flower color, my dear children, you had purple and white, or basically violet and white color flowers. Which one was dominant? Dominant one was purple. Which one was recessive? White one was recessive. The same thing went ahead. Plant height. The tall ones were dominant, whereas the dwarf ones were recessive. Now we're talking about seed color, seed shape. Remember, for everything there is two things. For example, flower color is there, and then you have flower position. Similarly, seed color is there, seed shape will be there. pod color is there, pod shape will be there. The only guy who is single over here is what? The plant height. Height does not have any other things out there. Now in seed color, my dear children, even though we eat green peas out there, but you know what? Yellow is the dominant trait. Okay, yellow one will be a dominant trait. Now if we talk about seed shape, round is the dominant trait, whereas wrinkled, do you eat wrinkled ones? No, we would not prefer that. Now if we talk about pod color, now when you go to the market and pick the peas, you look for the green pods, right? So the green color is dominant over here. In seed color, it is yellow, but in pod color, it is green, which is dominant, and yellow, which is recessive. If at all you talk about pod shape, the one which is full is what you choose. So inflated or full is dominant, and this one is constricted one is what? The recessive. Talking about the flower position, Baba, the flower position when it is in axial position means, suppose I have a tree over here, and from this tree's point where a branch is going to come off, I have a flower. This is the axial position. Now suppose there is a branch, at the tip there is a flower. This is the terminal position. Which one is dominant? Axial one is dominant. And to remember all of it, there is a small trick, and that is gravity. So if at all you want to remember which trait is dominant, just remember gravity. G stands for green. Okay, green in what? Pod color. R stands for round seed. A stands for axial flowers. V stands for violet flowers. I stands for inflated pods, that is the full pods. T stands for tall plants, and Y stands for yellow seeds. So these are the dominant traits out there. Did you get it? 7 pairs of contrasting trait. Is it clear? Can you give me a thumbs up if at all it is clear? And remember, this is just for the dominant character. Now once you know this, you can solve any question on the basis of this. But before I go ahead, I want you guys to give me a thumbs up, or else how would I know? So Zohan and Shubhas, thank you for giving me a thumbs up before. I saw it now. Understanding, not understanding. What's happening? Okay, so I'll go ahead, and by then I think you will be giving me a thumbs up. So why did you choose pea plants? That is also one very important thing. Okay, a plant. Thank you, Tanish. Thank you so much for giving that thumbs up. Okay, Subhash, I'm coming to that question. You know what? You have to wait until the end to find the answer for that question, because I have kept that question for the last. Okay, now listen to me carefully. Why did people believe Mendel's work? First of all, he started using math in biology. From tomorrow, if I bring some mathematics terms out there, 45% of this, 44% of this, you also will start believing me. Because why? We talk about the statistical analysis. So people started believing his work because he was the first one to use math in biology. And he didn't say that after doing experiments for 2 plants, I will be giving you results. No, he had a very large sample size and he went ahead with that. And he did repeated experiments. So many times he repeated his experiments until and unless he found that the results which were found is consistent. And also he chose plant with clear contrasting traits, and that is your garden pea, which you also call as Pisum sativa. Pisum sativa. Okay, and the year is already been mentioned. Please remember 1856 to 1863. This is super important. For 7 years he worked, and the plant had 7 pairs of contrasting character also. Now, going ahead with one question that I have in front of you. Everyone who understood the question, try to solve this. Which of the following is not a pair of opposing characteristics Mendel studied? This is from your question bank. Soon you will see question from the KCET pattern also. So I have mentioned the year for KCET, and the slide color would be red for KCET, so that you get an understanding. Both of it is being covered. Okay, can I try to solve this? Which of the following characteristics is not something that was studied by Mendel? What is the answer for this particular question? Axial and terminal flowers, full and constricted pods, green and yellow pods, pink and white flowers. Subhash says the answer for this question is D. Okay, what about others? Tanish, Zohan, Lavanya, Harshvardhan, Chandrakant, everyone out there, give me your answer. Okay, Subhash is like, no way, it's nothing apart from D. Yes, I got it. Anyone else with some other answer out there? Or shall I lock Subhash's option out there? Okay, Okay, okay. What is D? D says pink and white flowers. Arey Baba, there is no pink flower that Gregor Mendel studied. Okay, Gregor Mendel only had violet flowers. Violet and white flowers. Remember, gravity, V stands for violet. So over here, legend is there. Legend Soul Gaming is there, also saying D. Tanish also says D, which is why I love D. The answer for this question is actually D. All the other options are absolutely correct. So you get your marks in your 12th standard. Okay, that is your PU2. Yes, Nusrat. Nusrat is also super correct. Now going ahead with a KCET question on the same topic. Try to solve this. Which among the following characters selected by Mendel in a pea plant is a recessive character? Now Baba, tell me, which among the following character selected by Mendel is a recessive character? Inflated pod, green pod color, white flower, axillary flower. What do you think is the answer for this question? You have to find the recessive trait, okay? Now that should not fall under gravity. In the gravity stands for all the dominant traits. Think. Okay, Subhash says the answer for this question is C. Legend is also saying C. Okay, fine. What is C? C is white color. Okay. Amreen is there. Hi Amreen. Hi. Nusrat is saying C. And 33.9 Rihan is also there. Oh my God. Okay. Nikhil, ma'am, please say to upload chemistry board videos. Dear, definitely. We'll be doing that. Okay. Okay, Dinesh is there, Tanish is, every one of you are saying C, so your answer is C. White flower is actually the one which is a recessive character. Inflated is dominant, green pot color is also dominant, axillary flower is also dominant. Now, did you understand the flow? This is how we are going to study everything out there. Okay, so let me see what is the next thing. Next, we have the second concept, that is one gene inheritance, or you also call this as monohybrid cross. But before I go ahead with that, Name is also here. Awesome. You are also giving me correct answer. Dinesh also. Geeta also gave me the correct answer. By the way, guys, did you click on the like button? If at all you have not, click on the like button. And did you understand concept number 1? If concept number 1 is understood, give me a thumbs up. We will be going to concept number 2 then. White because dominant is violet and recessive is white. Exactly, Dinesh. Arey Baba, rocking. Awesome. Okay. You guys are super awesome. Do you know that? Children, can I go to concept number 2? So this is how it is going to be followed. Only when you give me thumbs up, here, I will understand that, okay, all my children understood this. Yes, Subhash said, yes, ma'am. Thank you so much. You can also say that also. Yes, great. Fine. Concept number 2 is one gene inheritance and monohybrid cross. Here also, it is super important. Listen to me carefully. What do you mean by monohybrid cross? Before I start off this, remember there is a 5-marker question in your PU2 boards that came from here. Represent schematically the result of one gene inheritance between pure tall pea plant and pure dwarf pea plant. How do you show this cross between pure tall pea plant and pure dwarf? This is how you are going to show that. Listen to me carefully. When I say that a plant is tall, okay, when I say the plant is tall, and then when I say it is pure breed tall, what I mean is, every time we have 2 chromosomes, basically a pair of chromosomes. Here, if I have an allele for height, I will also have one more allele on the other chromosome for height. Now, the alleles can be same like this, or it can be different also. If at all it is same, such as you can see over here, it is capital T here, capital T here, I will call this as pure breed, or I will call this as homozygous condition. Okay, when both the alleles are same, pure breed or homozygous. Similarly, here I see dwarf plant. This dwarf plant is also what? Pure breed, recessive, and it is also homozygous in condition, right? Both the small t alleles are there. Now, if we go ahead, from this guy, only one can come in a gamete. You know, in gamete, only half the number of chromosomes are going to go. Suppose there is a breakup that happened over here. Only among them, only one can go. Whatever one goes, my dear children, anyway, it is going to carry capital T because you do not have anything else. From here also, they will carry only small t. You do not have anything else. Now, suppose these 2 gametes came together. This gamete and this gamete came together. You again have a pair of chromosome. What is this pair of chromosome? If at all you see, my dear children, one is capital T and other one is small t. One is capital T, other one is small t. Over here, what is going to be the phenotype of the plant? How does the plant look? Phenotype means physical appearance. Capital T allele is dominant over small t, which is why this plant is going to look tall. But do you let me know whether it is pure breed or not? It is not going to be pure breed. Why? Do you see capital T over here? No, this is not pure breed. You call this as heterozygous condition. Hetero means others. Over here, both the gametes are different. Okay, so this is homogametic condition or homozygous condition. This is heterozygous condition. Now, in F1, this is the first generation that he got. All of the plants were tall. And they were tall like this. But Mendel did know. Mendel just saw that a plant is tall. He didn't know whether it was tall like this or it was tall like this. So, which is why he said that I want to do one more experiment. So, he went ahead and did one more experiment which goes like this. So here the parent is going to be capital T small t and capital T small t. From this only one can come in. So suppose it is capital and over here. Okay, just a second. Okay. So over here one came in. Either it can be capital T or it can be small t in a gamete. But only one can go in a gamete, right? Similarly here capital T can go or small t can go. Now imagine. Suppose this and this fused, this gamete and this gamete fused, you get capital T, capital T. Suppose this and that fused, you get capital T, small t. Suppose this and this fused, you get capital T, small t. Suppose this and this fused, you get small t, small t. Baba, if at all you see, this is tall because both of them are capital T, right? And this is also tall. Why? Because this capital T is dominating the small t. Because it's a dominant trait and that is a recessive trait. Similarly here, this is a dominant trait and the other one, my dear children, is going to be what? A recessive trait right there. And over here, this one is going to be dwarf because there is no capital T. So three of them, I say, three of them are going to be tall and one of them is going to be dwarf. 3:1. Someone was saying 3:1 and who was that? 3:1, someone was saying no. Where did that person go? Okay, I'm not able to see that person now. Anyways, 3:1 is what? The phenotypic ratio of F2 generation, filial 2 generation. What is phenotypic? How do we look? That ratio of the morphology of how do we look? The physical appearance. So 3 of them are tall, 1 is dwarf. What about genotypic ratio? Now genotypic ratio, I already, arre baba. Those of you are saying something else? Guys, check your answer. Oh my God, no, you're getting confused. Listen to me carefully. Genotypic ratio means what is their genotype. That is, what is the genetic combination. So 1 is capital T, only one is capital T among them. So I'll write 1, capital T, colon. 2 of them are capital T, small t. So I'll write 2, capital T, small t. 1 of them is small t. So I'll write 1, small t. This is going to be my genotypic ratio. This is going to be what? The genotypic ratio. Phenotypic, how we look. Genotypic, what is the genetic makeup? Can you see genotypic ratio? Can you see the genotype of an organism? For example, I am right in front of you. Can you see my genotype? No. But you can clearly see how do I look. You can clearly identify my phenotype, right? Yes. So what are you guys saying? Let me see that first. So Subhash is saying 1:2:1 is the genotypic ratio. Yeah, exactly, Subhash. Subhash also gave me the phenotypic ratio. Awesome. Lakshmi and Shais, hi dear. A very big hello. Lakshmi says, ma'am, what is the class duration? So I'm not sure about the class duration until and unless I finish this chapter once. There is no ending. But children, don't worry. I'm not going to torture you, okay? And Nusrat Baba, just check it once. I hope now it is super clear to you. Okay? Yes, Nusrat. Phenotypic is 3:1 and genotypic is 1:2:1. Only one small t is there. No, that's why. Cool. Dinesh, everyone. I hope everything is clear. Done. Oh, fine. So in one gene inheritance, you would have to explain all of it. If at all you are not explaining this, you will be losing your marks. So first explain about the F1 generation, how do we get capital T, small t, and then in F2 generation explain how capital T, small t is being crossed. That is the thing. You have to explain it fully, make it properly, you will be able to do it. Now, if at all you do not want to cross like this and this, you can just make a Punnett square. You can make a Punnett square like this. You can put capital T small t over here, capital T small t over here, and then do the crossing. This and this capital T, this and this capital T small t, this and this capital T small t, this and this small t small t. Always remember, this character, that is small t, will only show off if there is no dominant character. If there is no dominant character nearby it, then only it will be showing off. Okay, done? Chalo. Kudos. Now we are going to go ahead with one very important thing. So after Mendel did his experiment on the monohybrid cross, monohybrid means capital T, small t, it's a hybrid. It's not same, it's a hybrid. So that is why I say monohybrid cross. And why mono? Because over here I am only talking about one character, one trait, and that is what? Height. That is height. And in height, I have two alleles, capital T, small t. When I get this, I will call this as hybrid. Now, there are two laws that he gave after doing this one gene inheritance thing. One is law of dominance. He said that characters are controlled by discrete units known as factors. Okay, characters are controlled by discrete unit known as what? Factors. Now, factors occurs in pair. Yes, we have studied that factors occurs in pair. In a dissimilar pair of factor, one member of the pair dominates the other member. In a dissimilar pair of the factor, one is going to dominate over the other one. And the one that dominates is known as dominant gene, and the one that is not going to show up its character is known as what? Is going to be known as what? The recessive trait. Okay, so this is law of dominance. Always remember, after one gene inheritance, he gave one law. Now, after that, with the same experiment, he gave the next law also, that is law of segregation. And here, if you understand law of segregation, you have understood half of the chapter properly. Okay. Law of segregation means alleles do not blend. Both reappear unchanged in F2. Alleles do not blend. What do you mean by that? So basically, if at all you see over here, when I said in the first generation, you had capital T, small t, but the plants were tall. Do you think the small t allele went off crying that, oh my God, my character was not being shown? No, Baba, it was not like that. Okay, in the next time when he did the experiment, it was found that, yes, the small t came back again. So alleles, my dear children, are just going to be there. They are not going to blend up and show something else. It is just going to be intact. And he also said one more very important thing. What is that? Just give me a second, okay? And that is, during gamete formation, allele pairs separate, so each gamete gets only one allele. That means, I already told you, if suppose these are the chromosomes, here I have capital T, here I have small t, when gametes are going to be formed, these two guys are going to break up and only one guy is going to get into one gamete. Both of them cannot go into one gamete. Half of it is only going to go. Okay, so that is the thing. During gamete formation, allele pairs separate so that each gamete gets only one allele. Okay. Subhash, ma'am, a meaning of pure lines basically means, as I said, pure breed organisms where only capital T, small t are being crossed, and yeah, that's it. Okay. So yeah, we were here. Now, homozygous parents produced identical gametes. Homozygous parents mane this. Homozygous parents will be producing same type of gametes. Whatever you do, the gametes are going to be same. Now, heterozygous parents, my dear children, like them, are going to produce two type of gametes. Done? Ma'am, what are alleles? Acha. Okay, I can explain you that. Harshita, listen to me carefully, okay? And Subhash, nice PPT. Thank you. Sayesh also said nice PPT. Thank you, dear. Dinesh, I will recommend you not to write anything right now. When you get the PDF, you have the PDF, read through the PDF, that will be more than enough. Okay? Now, listen to me carefully for those of you who are confused within alleles. So see, I say in the chromosome, I have a gene over here. Gene means basically a segment of DNA that is going to code for something. Now, this gene that I have, this gene is for height. But in height, I have tall and I have dwarfness also. So that means gene will also be of separate types. Gene, if I had something for height, it will be also separate types. So one will be capital T, another will be small t. These separate things that I have, different forms of the genes that I have, are known as alleles. Now, alleles can be same like this, or alleles can be different like this. Here it is capital T, there it is small t. I hope it is understood. Okay? I hope this is clear, Harshita. So different forms of a gene is known as alleles. Cool? So I'm going ahead. I hope this is clear. We are seeing the first question out there. I don't know why sex determination is written over here. No, this is not. Okay. Try to solve this question. This is from KCET, okay? This is from KCET. I told you red background, that means KCET. In one of the hybridization experiments, a homozygous dominant parent and a homozygous recessive parent are crossed for a trait. Okay. Now, what do you observe over here? Dominant parent trait appears in both F1 and F2 generation, and the recessive parent trait appears only in F2. Dominant parent traits appear in F1 generation, and recessive parent trait appears only in F2 generation. Dominant parent traits appears in F2 generation, and recessive parent trait appears only in F1 generation. What would be your answer for this particular question? Anyone? Nusrat, if we work hard and study consistent and follow Deeksha Vedantu 0 to Hero series, can I score a rank below 1000 in KCET and more than 95% in PU2? So for KCET, definitely here in 0 to Hero series, we are planning to cover at least the high weightage chapters. Okay, so after the high weightage chapters, why we are not saying that we will be covering everything all at once is that, my dear, you would be having your boards, you will be having your pre-boards, that is the reason. Okay, sorry, you would be having your boards and practicals in between. So yes, we will be coming up with all the other things also, which is going to help you. This particular series will definitely make sure whatever we are teaching, whatever chapters we are teaching over here, you do not have to look again at these chapters for your KCET or your PU2 because it's done. Okay. Now, Lakshmi says the answer for this question is A. What is A? Dominant parent trait appears in F1 and F2 generation. Recessive parent trait appears only in F2 generation. Okay. Subhash says the answer for this question is A. Chiku is there. Hi, Chiku. Chiku says the answer for this question is B. Subhash again says A. Anyone else have any other different answers? Harshita also says the answer for this question is A. Okay. So since you guys are saying A, let's see A. Dominant parent trait. First of all, you are crossing what? You are crossing homozygous dominant parent. Homozygous dominant parent means it has to be same and it has to be dominant trait. So this is this. And you are crossing homozygous recessive parent. Homozygous recessive means the alleles are going to be like this and both of them has to be recessive. Now this is exactly that we did, the cross that we did. In F1 generation, what did you find? All of them were capital T small t. All of them were capital T small t. That means the dominant trait appeared in F1 generation. Only the dominant was appearing in the F1 generation. What about F2 generation? In F2 you saw that one was capital T, two of them were small t, and one of them was small t. So basically you again saw the dominant trait in F2 generation also, and for the first time you saw the recessive trait, that is dwarfness, that too in your F2 generation. For the first time. So this statement is correct, that is it appears, dominant trait appears in both F1 and F2 generation, whereas recessive parent traits appears only in F1 generation. So those of you said answer for this question is A, you guys are absolutely correct. Okay. Now next question is there. Before that, okay. So my dear children, those of you who have not taken up ABHYAS test series, I will recommend all of you to take that series for a complete KCET preparation. So previous year also we gave this particular course and so many thousands of children got the benefit of it. Thousands of children got entry into the best colleges out there, and it is definitely going to help you well as well. We had Prajwal with rank 171 and Kiran with rank 443, Deeksha with 693. You imagine all of their ranks are counted from 3 lakh children out there. So it's not a small deal at all, right? So there are 35 KCET tests, and apart from that there will be chapter-wise KCET PYQs. This is a new addition that we have put, but the pricing we have kept the same only. It is 1499. Chapter-wise PYQs is for everyone who is preparing for your PU2 boards also. So whenever you have time, once you're done with the chapter, go ahead and attempt the chapter-wise PYQs. You have a timer also, such that it gives you a real-time exam pattern. Now, the link to this will be there in the description box. Go check it out. Cool? Yes. Fine. Super. Now, I am going ahead. Let's see concept number 3, test cross and back cross. This is the next concept that we are having. So, the doubt over here is, this guy has a doubt. How did Mendel get to know if the plant is tall like this, capital T, or like this, that is capital T, small t, because both of them are going to be tall anyways. So that's when the use of test cross came into the picture, my dear children. And this question came in your PU2. What is the difference between back cross and your test cross? Back cross and your test cross. So how do you give the answer for this? What do you mean by test cross? Crossing an individual showing a dominant phenotype with its recessive parent. Didn't understand? I'll tell you. Just listen to me very, very carefully. So over here, suppose I have capital T, and I have capital T, small t, capital T, small t. This is tall, this is also tall. I have a plant and I have to find out whether it is like this or it is like this. How do I do that? I am going to cross it with one of the recessive parent. What I'm going to do? I'm going to cross it with one of the recessive parent, that is this. Recessive parent means homozygous recessive parent, okay? So what do? I'm going to make a Punnett square. From the recessive parent, I get alleles small t. From this, suppose I'm talking about this, okay? So here the alleles will be capital T. When I cross them, whatever I do, I am going to get capital T, small t only. That means all of them, my dear children, are still going to be tall. All of them are still going to be tall. On the other hand, if I cross this, let me see what is going to happen. So capital T, small t over here, I make the Punnett square. The recessive parent remains the same. When I cross this with this, I get capital T, small t. When I cross this with this, again capital T, small t. But when I cross this with this, I get small t. This with this, again small t. So what do I get to understand? Two of them are tall because you have the dominant trait. So two of them are tall, but two of them are going to be dwarf. So when I get progenies like them, I get to understand the plant is like this. When I get progenies all tall, I get to understand the plant is like this. This is known as what? Test cross. This is known as test cross. It is cross between F1 generation and a recessive parent. Yes, and it is there to find the genotype of the parent. It is there to find the genotype of the parent, whether it's like this or like this. So the exact definition which is there, it is like this, that is crossing an individual showing a dominant phenotype. What is dominant phenotype? That is tallness. Crossing an individual showing a dominant phenotype with its recessive parent, in the previous generation. That's all. And what is back cross? Crossing an F1 hybrid with either of its parent, dominant or recessive. So basically all test crosses that you see is a back cross. Back cross means you're just going back and crossing. Now you can cross with the dominant parent, you can cross with the recessive parent. Does not matter. You can cross with either of them. That is a back cross. But when you are specifically crossing with a recessive parent, you call this as a test cross. Okay, it is there to find the genotype. Now with that concept, we have a question. Try to find answer for this. Test cross in pea plant is what? Test cross in pea plant is a cross between F2 tall plant and a recessive parent, a cross between F2 dwarf plant and a recessive parent, a cross between F2 tall plant with a dominant parent, a cross between two F1 plants. What will be your answer for this particular question? Subhash, awesome dear. So here you do not have to be concerned about F1, okay, between F1 or the recessive parent. It's just that whatever organism you are having, you will be going back and crossing with the recessive parent. That's all. So what do you think is the answer for this question? A, B, C, D. A, B, C, D. So Nusrat says the answer for this question is A. Lakshmi also says is A. Subhash also is saying the answer for this particular question is A. Is it A? A means what? A cross between F2 tall plant and a recessive parent. Okay. Anyway, yes, Harshita is also saying the answer for this question is A. That means you guys, since you are all saying A, so definitely it has to be A, right? Number 100 is also saying A, okay. So a cross between F2 tall plant and a recessive parent is definitely a test cross. Why others are wrong? A cross between F2 dwarf plant. Why do you need to cross a dwarf plant with a recessive parent? No, Baba. Dwarf plant is always going to be like this only. We do not need to find the genotype. It has to be like this only. A cross between F2 tall plant with a dominant parent. If you cross it with a dominant parent, only dominant trait is going to be there, right? So this is again wrong. Cross between two F1 plants, arey Baba, we do not need it. So your only answer for this question would be what? A. Yes? Yes. Got it? Ma'am, it should be actually F1. So yes, I agree, in your NCERT it is given as F1. It should be considered as F1 only, but it's like, it's not necessarily need to be F1. But it is necessarily not needed to be F1, which is why it's a KCET question, okay? So I have not hampered it. This is from 2024. The question came exactly like this. So whenever you have a doubt in the phenotype, you do a back cross with a recessive parent, okay? Chalo. Now, the fourth concept. Can I go ahead with the fourth concept? When you write in the definition, if you want, you can go ahead with F1. Why? Because in your NCERT it is given like that, okay? But when you're applying for concepts, you can go like this also, if there is no other option which is saying F1. Cool. Shall we go ahead with the dihybrid cross experiment? If yes, can you give me a thumbs up? Ma'am, do ecosystem one shot. We'll do it. We are bringing a lot of things for you, so if at all you are not subscribed to the channel yet, subscribe to it. It is definitely going to help you out, Asha. Okay, thank you so much. So I'll go ahead. Subhash gave me a thumbs up. What do you mean by dihybrid cross? So until now, we were just considering one trait, one character, and that was what? Height. And in height, we spoke about two traits. One is tall and the other one is dwarf, right? So when we cross plants with, when we cross plants and when we get something like this, we say it's a monohybrid, since both the alleles are going to be different. Now when we talk about dihybrid cross, we are not going to study just one character, but we will see two character at once. And this is the most, what you call, trickiest part out there. Most of the children lose their marks over here. And this is the question that have again been put in your MQP1. Represent schematically the results of two gene interaction. Two gene interaction is basically dihybrid cross, taking seed shape and seed color in the pea plants. So let's see this particular question and let's get those marks over here. So yeah, the parents, junior Mendels, I hope all of you are super ready for this experiment. Let's go ahead, okay? So Baba, what I'm going to do is, I am going to take round and yellow seeded plant. This seed has the shape that is round. The color is what? Yellow. How do I represent this? I am taking pure breed round and yellow. That means round should be like this, capital R, and yellow should be like this. Okay. And I am crossing it with wrinkled and green plants. Wrinkled and green plants. And this is also pure breed. How do I show that? Wrinkled means, ayyo, why it is getting touched like that? Wrinkled means it will be smaller, and green means it will be small y. Remember, in seed color, yellow is dominant, and in seed shape, round is dominant, right? Now when we cross it, you need to find the gametes. How do you find the gametes over here? This is where most of us lose our marks, but it is actually easy. Look over here. So suppose I have 1, 2, and 3, 4 chromosome. Over here, I have alleles capital R. Over here, I have alleles capital Y. Agreed? Similarly, I have 4 chromosomes up over here. Here I have small r, and here I have small y. From this particular pair, that is capital R, from this particular pair, that is capital R, one is going to come over here. One is going to go down into a gamete. Suppose capital R went. From here, only one can go. Anyway, it's just capital Y that can go. That means I have a gamete with what? Capital R and capital Y. Either one of them. Okay. Similarly, I would be having small r and small y from them. Now, when I fuse it, what am I going to get? These are the gametes. There are going to be 4 gametes, but I'm just making one because it's all the same type of gametes that are going to be formed. When I fuse this, when I fuse the gametes, what do I find out? I find out capital R, small r, capital Y, small y. Capital R, small r, capital Y, small y. Now the picturization looks like this. In one gamete, you got capital R. From the other gamete, you got the small r chromosome. From one gamete, you got capital Y. From the other gamete, you got small y. Whichever is dominant, that will be overshadowing the smaller one and showing the effect. Which means all of them, my dear children, are going to be what? Round and yellow. Are going to be round and yellow. Oh my God, Subhash. Nusrat. Okay. Awesome. Okay, listen now. Okay, so this is round and yellow that I got in my first generation. Always remember, first generation will show dominant parental character over here. Now I am going to cross this. So this is where we need to be more attentive. So capital R, small r, capital Y, small y, capital R, small r, capital Y, small y. When I make chromosomes, now everything is going to look different. Capital R, small r, capital Y, small y. Now, if at all you look properly, from this pair only one can come. So, suppose I have taken capital R. From here only one can come, that is capital Y. So, this is one gamete, right? Now, from this pair again, suppose capital R came, but here small y came in. So, this is another gamete. From here small r came this time, and from here capital Y came this time. From here small r came this time, and from here small y came this time. So how many different type of gametes do I have? I have 4 type of gametes. I have how many? 4 different type of gametes from one parent. Similarly, from the other parent also I will have the same type of gametes. Right? Now if I put the 4 gametes here, 4 gametes here, and I try to fuse them, what will I find? When I try to fuse them, what? Just a second. Aish. If I zoom this, come on guys, don't do this to me. Okay. Think that, yeah, now it's okay. So when I fuse this with this, I get capital R and capital Y, capital Y. When I fuse this with this, I get something else. Similarly, how many combinations am I going to get? I am going to get 16 combinations out over here. Some can be repeated, some will be not repeated, some will be very unusual things also. So some repeated, some unusual. Okay, you will be finding unique over here, everything unique over here. In this manner, if at all, I try to find the phenotypic ratio, as in how many of them look round, how many of them look wrinkled and everything like that. I get a ratio which you guys were saying, and that is 9:3:3:1. Now this ratio is super important. Okay, this ratio is super important. Exactly, Subhash, non-parental. I'm coming to that. So 9 of them are going to show the dominant trait. What was the dominant trait? Ma'am, the dominant trait was round and yellow. 1 of them is going to show the recessive trait. What was the recessive trait? Ma'am, it was wrinkled and green. It was wrinkled and green. Okay, what about this 3:3? They are going to show non-parental character, something that was not there in the parents, something new. So round was with yellow until now, no. So let's make round with green this time. Wrinkled was with green, no. So let's make wrinkled with yellow this time. And that's how you remember 9:3:3:1 stands for what? These 2 things which are there, they are non-parental. That means out of 16, 3 plus 3, 6 of them are going to show non-parental character. And over here, my dear children, a very important law comes into action, and that is law of independent assortment. The 3rd law, the final law of Mendel. Law of what? Independent assortment, according to which he says that basically, if these alleles are for a character, that is what? Shape. And these alleles are for another character, that is what do you call the color, then both of them are going to assort or separate independently. Capital R does not say that I will only go with capital Y. It also goes with small y. And since it is giving new combination, that is why you see non-parental characters. If at all they were very specific that capital R will only go with capital Y, you will not see all these special characters out there. And that is law of independent assortment. And that is something that I have written over here. So did Gregor Mendel find something unique? By the way, say hi to Gregor Mendel, everyone. So this is Gregor Mendel, sir. And what unique thing that he find? He found the law of independent assortment with the dihybrid cross experiment. With the dihybrid cross experiment, he said that when 2 pairs of traits as in 2 traits, 2 pairs of traits as in one for what do you call the shape, other for the color, whatever you're taking, segregation of one pair of character is independent of other pair of characters. Segregation of one pair of character, that is the color, will be different from the segregation of other pair of character. Capital R can go with capital E, move together. This is not going to happen at all. Okay? Good? Ma'am, how to study biology? This is how we study biology. Understand the concept, solve the questions. Understand the concept, solve the question. That is how we study biology. If at all you just go and read your NCERT and do not solve the questions, you will lose your marks. Okay, because you know what, different type of question comes every single time. So you cannot expect anything. Okay, so this is a KCET question that have come. Everyone pay your attention over here. Here you just not have to use the dihybrid cross thing, but also what we studied in the one gene inheritance, that is the monohybrid cross. So in a dihybrid cross between a true breeding round yellow seeded and true breeding wrinkled green seeded plant, the ratio of segregation of round and wrinkled seed traits in F2 generation is what? The ratio of segregation of round and wrinkled seed traits. Here you are not talking about the other yellow or green trait which is being mentioned. Even though you are crossing this with this, they are asking you how many, okay, how many of them are going to show round and wrinkled trait. That is the question. So what is your answer for this particular question? What will be your answer for this particular question? Subhash says the answer for this question is C. What about others? Any different answers? Or you also have the same answer? This is what do you call a pretty advanced question. Okay, this is a pretty advanced question that you have. So you need to think and then only tell the answer for this, okay? I'm talking about round and wrinkled. Just round and wrinkled. So if I go back here, 9 of them were round and yellow, 1 of them were wrinkled and green. Over here, if at all I see round. How many of them are round? 9 are round, 3 of them are round. 9 plus 3 is equals to what? 9 plus 3 is equals to what? 12. What about wrinkled? 3 and 1. 3 and 1. If I get 12:4 ratio and if I do this, I get 3:1. And you do not even need to do that also. You do not even need to do that. Just understand, when you were talking about only one trait, monohybrid cross, wasn't the ratio between the tall and the dwarf 3:1? The same thing is being followed over here. The same thing is going to be followed over here, which is why I said whatever you studied previously is going to play an important role in this particular question. So your answer for this question is going to be 3:1, my dear children. Did you understand this? Everyone who is watching, yes, understood how it is 3:1? Because you're just talking about the dominant and the recessive trait, and this is just one character that you're talking about, that is the shape. Okay? Chandra, Baba, Chandrakant, dear, just check through. The answer for this question is B. If at all you have understood it, just let me know. Okay? Now there is one more question that I want to show you, and that is this. Try to solve this question. This is also a little more advanced. This is again a KCET question that is seen. A pure breeding pea plant with round and yellow seed was crossed with a pea plant having wrinkled and green seed. Again, round and yellow seed with what? Pure breed, wrinkled and green seed. On selfing of F1 hybrid, that means when F1 hybrid were crossed, okay, 64 progenies were obtained in the F2 generation. How many? 64 progenies, my dear children. Find out the number of F2 progenies showing non-parental character. Find out the ones which are showing non-parental characters. Out of 64, how many of them show the non-parental character? What do you think is the answer for this question? Chandan, okay. Now I think that, Chandrakant, I hope that is clear. You have given me the answer, that is B. Now we are solving this question, my dear children. Can you try to solve this question, everyone? What will be the answer for this particular question? Come on, come on, come on. Everyone who is watching this video, even if you give me wrong answers, is totally fine. But just attempt. Attempting is super important over here. Okay, Subhash says the answer for this question is C. Okay, fine. What about others who are watching this video? What do you think? Out of 64 progenies, how many of them are going to show non-parental character? So we had a ratio 9:3:1. 9:3:3:1. One of them round and yellow, the dominant trait, right? Okay. Legend says the answer for this question is B. Okay. Chandrakant also says the answer for this question is B. Fine. Let's see. So 9 of them round and yellow, 1 of them wrinkled and green, and these two are the non-parental character. What is a non-parental character? If round was with yellow, then round is with green now. And if wrinkled was with green, it is with yellow now, right? So these are the non-parental character. That is 3 plus 3, 6 of them. 3 plus 3, 6 of them. So 6 out of 16 are non-parental, and over here you are saying that 64 progenies are there. So you do this calculation. You do this calculation, 6 by 16 into 64, and when you do this, you get the answer to be 6 into 4 is equals to what? A, 24. What is it? A, 24. Got it? So I get options as D. Nusrat said D. Dinesh also said, was asking D. The answer for this question is what? A. Got it? I know it's a bit tricky out there, but you guys are learning something. The same mistake probably you might have done on the day of your examination, but you're not doing that. Be grateful for that. Okay, you're learning something new, and even if you gave the wrong answers, it's totally fine in that way. Got it? Clear? If this concept is clear, can you give me a thumbs up? I think because we are going to go ahead with the next concept. Yes. So before I go to concept number 5, if at all this concept was clear, can everyone give me a thumbs up right now? And why is this class, not super energetic? So when I came one day before examination, all of you were like, dar, answers, all of you super energetic, all of you shared this video with your friends also, but now I see less energy. Why? Because your pre-boards are just gonna come, you have days, that's why. Well, my dear children, be energetic. That's how we felt refreshed, no? Otherwise, if we study like this, oh my God, I have to study today, I don't want to study. If you do like that, you will get bored at the end, right? So don't do that. And just imagine me. Whenever you think of yourself, just imagine me. I am in a closed room, I am talking to myself basically. And if I have this much amount of energy, so I expect a lot of energy from you guys also, no? So just have lots of energy and share this video with lots of your friends also. Okay? Done? Deal. So from this point onwards, that is concept number 5 onwards, you guys should be having high amount of energy. Exceptions to Mendel's law is concept number 5, and what are the exceptions? Let's see what are the exceptions. Incomplete dominance is an exception. Over here in your MQP2 there's a question. Schematically represent incomplete dominance by taking inheritance of flower color in Snapdragon as an example. This is what? This is what? Basically a exception of Mendel's law, because whatever Mendel taught, somewhere it was getting wrong over here. Why? Let's see that. So this is incomplete dominance shown in Snapdragon or Antirrhinum majus, my dear children. What is Subhash saying? Mendel was not having communication, no phone, so he was using only lab. Exactly. Okay, so he crossed red colored flowered plants with white ones. So capital R is for red and small r is for white. So my dear children, what do we all expect? Snapdragon flowers, or you call it Antirrhinum majus flowers. We expect that when we cross this, anyway, whenever we are going to cross this, all of them are going to be capital R, small r. And if all of them are going to be capital R, small r, capital R stands for red. So this is dominant and this will be showing red color. But my God, everyone was shocked, my dear children. Why? Because here it was not showing red color. Instead, it was showing an intermediate color, and that is pink. And that was against law of dominance. Law of dominance said in a pair of alleles, one allele is going to be dominant. Arey Baba, this is wrong over here. This allele is not completely dominant, and that is about incomplete dominance. And since they were shocked, they did one more time. They crossed one more time. So over here, they took capital R, small r and capital R, small r and crossed it again. So the alleles are capital R, small r, capital R, small r. If you cross this with this, you get capital R, capital R. If you cross this with this, you get capital R, small r. This with this, again capital R, small r. This with this, small r, small r. If at all you get a combination like this, Baba, what is going to happen? Exactly, Chandrakant, you were saying pink. Yeah, right. So if at all you get a combination like this, when capital R comes together, it is red only. It is red. So they were like, oh my God, finally. Their void may somehow have problem out there. But when they saw this, capital R, small r again was pink. This was also pink. And small r was white only. So then it was deduced that here some problem is there when capital or small are coming. Probably it is going to be incompletely dominant. It is going to be incompletely dominant out there. And that exactly happened. And that's why we say there's a law of incomplete dominance, where the genotypic and phenotypic ratio are also going to be a bit different. So if at all you see the, the phenotypic ratio. One is red, two of them are pink, and one is white. So the phenotypic ratio, my dear children, is 1:2:1. One red, one white, and two pink. What about the genotypic ratio? Genotypic ratio is going to be the same like how we had in the F1 generation of one gene inheritance. It was, genotypic ratio is what? 1:2:1. One of them capital R, two of them capital R, smaller, and one of them smaller. Got it? So this is incomplete dominance, super important. Ma'am, phenotype and genotype same, exactly. In incomplete dominance, the phenotype and genotype is same. Subhash, an intermediate is being formed, exactly. Chandrakant also gave me the ratio 1:2:1. Awesome. You guys are super awesome. Okay? So this is about incomplete dominance, and let's see if at all do we have any question on the basis of it. So my dear children, you can make the Punnett square and try to explain, okay? Nusrat, can I get 70 on 70 in biology through 0 to hero series? You can get 70 on 70 in biology. Okay? Yes. Let's go ahead. So the genotypic ratio of incomplete dominance, my dear children, everyone, this is a KCET question. Are you able to solve this KCET question now? The genotypic ratio of incomplete dominance is what? And this is the first exception also. Please remember, this is the first exception of Mendel's law. Subhash says the answer for this question is B. What about others? Nusrat? What is the answer for this particular question? Chandrakant also says it's B. Nagaraj is here. Hello, Nagaraj. Okay, Nagaraj also says it's B. Awesome. Nusrat also says it's B. Now, since all of you are saying it's B, I'll log B. You guys understood the concept super clear, right? Now, going ahead with the second exception that we have, that is multiple allelism. Mendel said usually one gene, if at all you're talking about height, to have two alleles, capital T, small t. But it was found that there is a concept which is known as multiple allelism. Multiple alleles can be there, and that was found in the case of blood group. There was an allele known as IA, IB, and IO. 3 alleles are there, my dear children. So when there are multiple alleles governing a character, that is your blood group, it is known as multiple allelism. If multiple alleles are basically governing a character, you call this as multiple allelism. And this is a second exception. Regarding this only, blood group is also an example of something else, which is the next exception, the third exception, and that is codominance. So you all have studied, one is dominant, other one is recessive. Both of them cannot be equally dominant, right? But in the case of blood groups, my dear children, 2 alleles can be equally dominant. I'll tell you that, but before that, we need to read out this. Okay. Multiple alleles follow law of dominance. It can. Multiple alleles is totally different from law of dominance. Multiple alleles means there are multiple alleles. That's all. There are multiple alleles. Now, some can show dominance, some can show codominance. Anything can happen. Now, Garaj, a very good afternoon, dear. Okay. So, we were solving this? Yeah. So, before I go about that, just understand the basic thing about red blood cells. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface like this. Okay, and the kind of sugar is controlled by the gene. Okay, what sugar will be there will be controlled by the gene. It can be IA, it can be IB, it can be IO. Okay, these genes control what sugars are going to be there over there. Okay, the gene I has 3 alleles. Gene I which is there, it has 3 alleles. I told you about the 3 alleles. Now we are going to go about the basics of what exactly is codominance. So my dear children, over here you have to think of few scenarios, which is why I am going to make a chart like this. Suppose there is A blood group, there is B blood group, there is AB blood group, and there is O blood group. Okay, now listen to me carefully, it is super important. Now I'll shift the AB to last and O here. Okay, if someone needs to have A blood group. How many of you are A blood group people? How many of you are A blood group people? Any idea? Any idea? Chalo. So over here, for being A blood group, you should either have something like this, where two alleles should be like homozygous in homozygous condition, or it can be like this, where one allele is capital A and the other allele is O. Why is it like this? Because, my dear children, A is dominant over O. A is dominant over O. So in this condition, you can have A blood group. B blood group, both of these alleles have to be B, otherwise there is an option, one is B and the other one is O. So when I say that I have A blood group, I can be either of this. When you say that you are B blood group, then you can be either of this. We never know. Okay, A positive, Nusrat says A positive. Me also, I am also A positive. I do not know my blood group, it's very easy to find the blood group. Okay, now coming on. What about O blood group? You have seen that B is also dominant over O, A is also dominant over O. How do you think will a person have O blood group? What should be the condition be? The condition is super easy. Okay, it will be like this. Since everyone is dominant, it can only appear in recessive condition. Only appearing in recessive condition. So for O blood group to come, either of the alleles should be OO. That means from two of the parents, I should get one allele of each of them. Now, what about AB? This is where codominance come. AB means one allele has to be A and other allele has to be B. In this condition, both of them are going to express themselves, and that is exactly what you mean by codominance. This is codominance. Both of them are equally dominant. So if you have AB blood group, that means you have two alleles which are both saying that I am dominant. Okay? Got it? Yes, I. So sometimes this IOO can also be mentioned like this also, is the same thing. Allele IO can be mentioned like this also. Rishi is here. Hi Rishi. Winter Luna. Hello. I have a different pre-board. Oh my God. Okay, so listen to me. It means follows law of dominance. See, multiple alleles means there are multiple alleles. Now some, as I said, can be codominant, some are not codominant, right? Here you see dominance is there. So it does not matter, it's about what you are being talking about. In the case of blood group, there's an example like that. There are multiple things where other things are happening. Okay, so I hope this is clear. This is what? Codominance. So if at all you see the last part, when IA and IB are present together, they both express their own type of sugars. This is because of codominance. Now this part was super important, which is why you have so many questions around it. Try to solve the first question from KCET and then we will go ahead. Okay, then we will go ahead. The genotypes of a husband and a wife are IAIB and IAIO. Among the blood types of their children, how many different genotypes and phenotypes are possible? That means genotype of a husband is this and the wife is what? This. So they are asking you, how many new genotypes and phenotypes can you find? What do you think is the answer for this particular question? Come on. Easy, simple. And everyone who is watching this video, share it with your friends. I'm telling you again, share it with your friends. Let them also watch it. Let them also understand the concept. Let them also score full on full. Yes? So Subhash says the answer for this question is C. Is it C? Everyone, try to cross them. If I cross this with this, I get IA. If I cross this with this, I get IO. If I cross this with this, I get IB. If I cross this with this, I get IO. How many genotypes are there? Friends are sleeping, ma'am. Wake them up, tell them. This is super important. Nagaraj says C. Okay. Subhas also says C. Fine. Don't spam. So C means 4 phenotypes and 3 genotypes. Let me see 3 genotypes are there. Genotypes mean genetic makeup. This is a unique genetic makeup, IAIA. IAIO is there. This is seen for the first time. So this is another genotype. First genotype, second genotype. Third one is IAIB. This is the 4th genotype. IBIO is the, This is the third genotype, and this is going to be the fourth genotype. This is all unique, my dear. None of them are going to be repeated, right? So how many genotypes do I see? I see 4 genotypes. I see 4 genotypes. What about phenotypes now? Let me see phenotypes. IA means A blood group. Both of them are AA. That means A blood group. A and O means again A is dominant over O. Again A blood group only. A and B, both of them are there. That means AB blood group because they are co-dominant. B and O is there. That means B is dominant over O. That means B blood group. A is repeated twice, which is why I'll just count it once. I will count it just once. So 1A, 1AB, and 1B. How many different types of phenotypes do you see? You see 3 phenotypes. You see 3 phenotypes. And with this, what is your answer for this question? Your answer would be B. Who said C? Who said C? Arre. Friends are enjoying festival. Okay. That is actually lol. Okay. So they should be enjoying festival. I should not be someone who should, what do you call, come in between. But sadly, I am in between. So my dear children, anyway, those of you who are attending this live, let's enjoy and attend it. Okay. So over here, the answer for this question is B. So those of you who said C, wrong. Okay, there's 4 genotypes and 3 phenotypes. Correct? Understood? Don't do the mistakes. Silly mistakes would be there. Don't do that. Now going ahead with the next question that we have is again based on AB blood group. In AB blood group, you have so many questions. What is a possible blood group of the children whose parents are heterozygous for A and B blood groups? Now tell me the answer for this question, and then I'll understand that everyone basically has understood the concept well. What is the possible blood group of children whose parents are heterozygous for A and B blood group? It is A blood group only, it is B blood group only, but they are heterozygous. What do you mean by heterozygous condition? Alleles should not be same. If alleles should not be same and then it has to be A blood group, then what do you think alleles will be? B. Subhash is saying B, B, B. Now if at all it is not B, let me see. Okay, anyway, I won't do anything. So heterozygous for A means it is I, A and I, O. This is heterozygous condition. And this is heterozygous B, means I, B, I, O. B blood group will be there. Why? Because B is dominant. A blood group will be there. Why? Because A is dominant. Now when I cross this, I get I, A, I, B. That means AB blood group is there. Agreed? Done. Now when I cross this with this, I get I, A, I, O. A is dominant over O, so A blood group is there. When I cross this with this, I get I, B, I, O. B is dominant over O, so B blood group. Arey wow, sub blood, every blood group is there. Now last is I, O, I, O. O is there, that means O blood group. So basically, what is a blood group that I cannot find? Nothing. I can find all the blood groups. So A, B, O, everything is there. So your answer for this question is B. Now let me see who all gave me the correct answer. Yes, now everyone. Subhash also perfect. Nagaraj, Silini, hi dear. Zohin is there. Hi dear. Dinesh Baba, check once more. 100% option B. B for butterfly. Okay, perfect. Winter Luna also. Chandrakant also. Zohin also. Awesome. Now before you change your answers, I'll go to the next question. Okay, the next question would be this one. Try to solve this question. A man with blood group A marries a woman having blood group B. The maximum possible blood groups among their progenies are. Now this question and the other question is similar type of question. Nusrat also gave me the answer for that question. It is B. Awesome, Nusrat. Awesome. Okay, try to find this question. Again, a man with blood group A marries a woman having blood group B. The maximum possible blood groups among the progenies are. They didn't mention heterozygous, homozygous. You have been asked about the maximum blood groups possible. What will be your answer for this question? Think and answer. If I take it to be just normal IA and IB, I won't find the maximal blood groups, right? I have to think extreme. So Nagara says answer is A. Winter Luna also says the answer for this question is A. Nutras also says it's A. Chandrakant also it's A. Guys, are you sure about A? Are you sure about A? Silini also says it's A. Since you guys are saying it's A, I'll lock A. Exactly. This question And this question is same question, Baba. What is the possible blood group of children having heterozygous A and B blood groups? Okay, if you do this cross, then only you'll get the maximum number of blood groups. We just saw that. The same thing is happening over here also. The same thing. If at all you consider A blood group as IAIO, and if you consider B blood group as IBIO, then only we can find maximal, what do you call, things like that, right? So same question. This came in 2020 and this came in 2021. 1 year gap, same question in KCET in different format. Okay, now you understand why I say this topic is important. Now this is from your MQP1. Try to solve this question. This is for your PU2. So 0 to hero series is for your PU2 and your KCET, and I am proving it that to you right now because we are discussing both of them, right? So try to solve this question. Progenies with AB and O blood groups are not possible from which of the following mating parents? Progenies with AB and O blood groups are not possible from which of the following mating parents? By the way, whoever is watching this, have you clicked on the like button? If at all not, go click on the like button right now. Right now. Super important. Subhash says the answer for this question is A. Subhash, progenies with AB and O should not be coming. Progenies with AB and O blood group should not be coming. That is the thing over here. So you have to think very carefully and then only tell the answer. If I take this. Now, you guys have to see this. So, IA and this is IB. This is IO and this is IO. O blood group means IO, AB means A and B, right? When I cross this, when I cross this with this, I get IA, IO, that means A blood group. If when I cross this with this, I get IA, IO, that means again A blood group. This with this, IB, IO, that means B blood group. This with this, IB, IO, that means B blood group. Do I get AB and O? No, which is why Subhash was correct. Okay, the answer for this question is A. And it is a simple way to, you do not need to solve all of it. Just understand, you do not need AB blood group, right? If at all you do not need AB blood group, that means A should come from one side and B should come from other side. That's when you get IA, IB, right? So here, if they are in opposite direction, do not pick that option. For example, here you have A, here you have B also, wrong. So we do not want this. Here you have A, here you have B, again wrong. Here you have A, here you have B, again wrong. Don't calculate all these things. Okay, so your answer for this question is going to be A. Got it? Baba. Select one. A, B, C, D. Okay. Got it? Clear? Jaggas. Ma'am, for 1 second my heartbeat was lubbed up. It happens. I like giving some shock in between. Now, everyone, my dear, I told first. Yes, you told first. Sileni, awesome. Okay, Sileni and Dinesh, Nusrat, Winterluna, Nagaraj, everyone, Subhash, everyone are rocking the floor right now. So let's go ahead. Let's see the fourth exception. So we are dealing with the exceptions of Mendel's law. That is our concept right now. We read about incomplete dominance. That is one exception. Second exception, multiple allelism. Okay. Third exception was what? Was what? Codominance. And now over here your fourth exception is polygyny. Polygyny is written somewhere else in your NCERT, but please understand this is also an exception. Why do I say so? But before that, me and all of them who are over here, we look so different, right? Why? Why do we look so different? In skin shade. In skin shade we all look so different. The reason is that polygyny is coming into action. So what do you mean by polygyny? So according to Mendel, he said that one gene, one character. Okay, one gene, basically one character. That means he said capital T or capital T small t over here gives tallness, and that's all, right? One gene on one location that gives a role for the character. But according to polygyny, it means that if at all you have multiple genes over here. Suppose I say it's A, it's A, it's B, it's B, it's C, it's C. All of them are going to code for something, and that is going to decide my skin color, or basically my height. This is known as polygenic inheritance. Multiple genes coding for one character is polygenic inheritance. Now in this particular condition, I say the genotype is producing some protein out there. Okay, so it is known as darkest skin tone. Now suppose I want to make it a bit lighter. Suppose I want to make it a bit lighter, I will add small a over here. So capital A, small a, capital B, capital B, capital C, capital C. This is a bit more lighter than the previous one. So in that sense, which one will be having the lightest skin tone? If I want to have the lightest skin tone, then my dear children, over here, everything will be recessive. Small b, small c, small c. That means A, B, B, C, C. This will give me the lightest skin color because less amount of protein, or whatever protein is being produced, the thing is going to be less. This is known as polygenic inheritance. Multiple genes code for a single character. Example is skin color. Example is also your height. Example is height and skin color, which is why we do not have similar type of skin color. Everyone have a different genotypic combination coding for different skin color. Everyone have a different genotypic combination coding for different height also. Got it? Yes, Nagaraj. Yes, Subhash. So since you have got this, since you have understood this, let's go ahead with one very important question, that is this. This is from KCET. Choose the possible genotypes responsible for the lightest skin color in human beings. Lightest skin color in human beings. So whenever I say that this is for KCET, does not mean that you do not need to, you need to close your ears if at all you're preparing for PU2. Remember, the KCET questions are also taken for your PU2 MCQs. So please solve them. They are super important. Okay. Tell me the answer. Choose the possible genotypes responsible for the lightest skin color. Subhash says the answer for this question is C. What about others? What about others who are watching this video? Zohin says C. Winter Luna says C. Nagaraj also says C. Chandrakant says C. Okay. Come on. Okay. Since everyone is saying C, let's lock C. C is what going to give me the lightest skin color. I told you why. Because all of them are recessive in nature. Polygyny done. That is your fourth exception. Now your fifth, sorry, before I go to that, there is a PU2 based question also that has come. Inheritance of skin color in humans show different phenotypes. Inheritance of skin color in humans show different phenotypes. Why? Because skin color in humans is an example of polygenic inheritance, where multiple genes contribute to the phenotype, leading to a range of skin color variation. This is what you need to write. Okay? Fine. Now, I already explained you this, so we do not need to talk about it. Please remember, skin color and human height, both are the examples. Okay? Now, before, just a second. Okay. Fifth exception is pleiotropy. Fifth exception that we have is what? Pleiotropy. What do you mean by pleiotropy? Now, pleiotropy is ulta of polygyny. Polygyny, you studied multiple genes, one character, but here, one gene is responsible for multiple character. Did Mendel say this? No, Mendel said one gene, one character. But here, one gene, multiple character. And the best example that we have is PKU exactly. And that is what? Phenylketonuria. You might have heard about this disease. It's a disorder. It's a genetic disorder that we have. And basically, what exactly happens in this? One gene, that is the gene coding for the enzyme phenylalanine hydroxylase. Okay, phenylalanine hydroxylase is going to be affected. This particular gene is going to be affected. It is not going to be produced. And when this is not going to be produced, two things are going to happen. Okay, the first thing, phenylalanine, which is an amino acid, it is not going to be converted to tyrosine. Baba, if it is not going to convert it to tyrosine, you do not know what's the importance of tyrosine. Tyrosine is super important for our skin pigmentation, hair pigmentation, because it helps in the formation of melanin. And if melanin is not there, what will you be having? You will be having reduced skin and hair pigmentation. That means it will be light in color. Your hair and your skin will be turning gray and whitish in color. Why? Because melanin is not being produced. Why? Because basically tyrosine was not being formed. The conversion is not happening. So this is one character being affected. Okay, this is one character being affected. The second one which is being affected is that the phenylalanine, since it is not being converted, it accumulates in our body in the form of phenyl pyruvic acid. Okay, it accumulates in the form of phenyl pyruvic acid. Phenyl pyruvic acid. And now when it's accumulated in our body, it leads to mental retardation as well. It leads to mental retardation. So two problems. One is reduced pigmentation and the other one is mental retardation caused because of the same thing, because one particular gene was not working. Did you get it? Tyrosine now. Just guys, stick to NCERT, that's all. Stick to NCERT, do not make it, wait. There is no reduction in your hair, okay? There is no reduction in your hair. Don't think like that. This is reduction of pigmentation is going to be reduced, okay? Hair is there, but everything is going to be pigmented less. That means it might be gray in color. So reduction of hair pigmentation, not reduction of hair, okay? Hair will be there. Cool? So that is the thing. So when a gene influences different metabolic pathways or phenotypes, this is known as pleiotropy. Phenylketonuria is an example caused by mutation in phenylalanine hydroxylase. The phenylalanine hydroxylase enzyme converts phenylalanine to what? Tyrosine, and its absence, phenylalanine will be converting to phenyl pyruvic acid, causing what? Mental retardation. The second character it hampers is reduction in hair and skin pigmentation. Reduction in hair and skin pigmentation. So read both of them together, okay? Hair and skin pigmentation. Fine? Got it? Yes. Only color. Yes. Subhash, different level only. Okay, so yes, that is about pleiotropy, and let me tell you, everyone who is watching this video understand in KCET, pleiotropy related questions have been asked so many times. So 5 exceptions we studied, and the 5 exceptions that were there for Mendel was incomplete dominance, then we studied about multiple allelism, codominance, pleiotropy and polygeny, and now we are going to solve some questions related to it. Let's start solving this question, okay? Yes, it's an inborn error mechanism, inborn error in your metabolism. Okay, you are born with it. We'll study about it when we study Mendel's disorders. So when a single gene exhibits multiple phenotypic expression, the phenomenon is called. When a single gene exhibits multiple phenotypic expression, multiple physical characters out there, or multiple things out there, a single gene, what do you call this phenomenon as? It's an easy question. Try to solve this question. C. Chandrakant, Subhash says the answer is C. Yes, answer is what? C. Single gene. When the term single gene comes in, just understand you're talking about that. Yes, Zohin. Yes, Winter Luna. Everyone. Awesome. Nagaraj, yes. Nagaraj said, okay, ma'am, before. Yes. So Nagaraj understood what I was trying to say. And yes. Shall we go ahead? Exactly, Subhash. So the next question related to pleiotropy goes like this. And also this is related to, what do you say? This is also related to your AB blood group. So please make sure, if you have understood the concept, all of it together, you will be able to understand this also. Generally, a gene regulates a trait, but sometimes one gene has effect on multiple traits. Okay, that is one thing. That is first statement. Second statement, the trait AB blood group of man is regulated by one dominant allele and another recessive allele. Hence, it is known as codominant. What do you think is the answer for this question? Both the statements are correct. Statement 1 is correct. Statement 2 is correct. Heading itself tells the answer. Yes. But still, Try. Come on, champions. What is the answer for this particular question? Generally, a gene regulates a trait, but sometimes one gene has effect on multiple traits. This is what? Pleiotropy. This statement is correct. Statement 2. A trait AB blood group of man is regulated by one dominant and another recessive allele. Is it? And hence it is known as codominant. Codominant means what? Both of them are equally dominant. There, one cannot be dominant and one can be recessive. So this is wrong. If you are putting codominant over here, this is going to be wrong. And A and B, both of them are actually showing codominance, where both of them are equally dominant. There is no role of recessive allele out there. You get it? So this statement is going to be wrong. So what is the answer for this particular question? What will be your answer for this particular question? Now you're saying D. Winter, Luna, Chandrakant, Subhash, Dinesh, everyone? Yes, the answer for this question is what? D. Statement 1 is correct. So were you waiting for me to answer? Okay, it is D. Cool. Let's go with the next concept or next question. Try to solve this question. ABO blood type in man is an example of. ABO blood type in man is an example of pleiotropy, incomplete dominance, codominance, multiple allelism. D for donkey. Okay. Nagaraj is back. Hi, Nagaraj. Okay. ABO blood group, blood type in man is an example for. Come on. Pleiotropy, incomplete dominance, codominance, multiple allelism. Subhash says the answer for this question is B. A, C and D only. Okay. What about others? Nagaraj says C. Winter Luna says C. C and D only. Okay. Subhash has now changed the answer to C. Okay. Winter Luna also says C. Okay. Children, the answer for this question is going to be C. Why C? Pleiotropy, Baba. Pleiotropy means what? Pleiotropy means what? We studied it. One gene, multiple effects, right? Here, ABO gene does not have any multiple effects rather than showing off the sugars which are there, coding for the sugars. So this is not going to be the one. Incomplete dominance, they do not show that. Codominance is something that they show when IA and IB come together. And they also show example of multiple allelism. So your only answer for this question would be C and D. Okay, so everyone who gave me the answer as, C are correct. Okay, Dinesh, Baba, why B? Why B? Is there something else that I am missing somewhere? Or I hope you have got it. Dinesh, Baba, got it? Shall we go ahead with the next thing? Concept number 6 is what I'm going to get ahead into. If you are clear with all the concepts that we have done so far, can you give me a thumbs up? Everyone, can you give me a thumbs up if we can go ahead with rediscovery of Mendel's work? So until now, Mendel was being celebrated. Okay, until few time Mendel was being celebrated. Then we saw that basically what happened, Mendel's law was basically scrutinized by so many people. They said that there are some exceptions to it. So I will not believe Mendel's work anymore. Okay, I am not going to believe Mendel's work anymore. And that's when lots of things happened. So are we ready to go ahead with it? If yes, can you give me a thumbs up, everyone? By then I'll drink some water also. Okay, thank you, Subhash. Nagaraj, Baba, yeah, okay, see, got it. By the way, everyone who is watching this video, have you drank water? Please drink some water, okay, and stay hydrated, then only you will have energy. Cool? Chalo. We'll do rediscovery of Mendel's work. So before I go ahead with that, you need to understand what went wrong. Why was Mendel's work, Mendel Uncle, why you discovered genetics? Exactly, this is also what I asked when I was in your grade. Okay, so, yes, something went wrong. What was that something that went wrong? Communication was difficult, so his work wasn't widely known. That was one very important thing. There was no communication, that means no appreciation, nothing is there. Okay. Second thing, his idea of discrete factors and no blending was not accepted. That means capital T, small t. There is no blending Both of them appear as such. There is no middle character. That was also not accepted because people showed that incomplete dominance was also there. Using mathematics in biology was unusual and not accepted. Okay, people, math and bio, oh my god, that's crazy. People who were appreciating that, they were like, oh no, this is crazy, I don't understand anything. Okay, that was a thing. Now the fourth thing is that he had no physical proof of what genes were or how they looked. Okay, he spoke about alleles, he spoke about all these things, he called them factors. He didn't see these genes, he just know that organism had it. You just imagine his brain. Oh my god, without seeing all this thing, he was going on with a lot of things. He must be a proated, right? Yes, he was a proated, which is exactly why, even though he had a downfall in his life, similar to us, we all have downfalls in our life. In our preparatory examination, some of us might have not answered really well, right? We might have not got great marks, but then we will be also having something that is known as a twist. And then came a twist. And what was the twist over here? That his work, but after 37 years, that is also need to be mentioned. So he worked in 1856 to 1863, and after 37 years came the twist. For you guys, don't worry, it won't be that long. This year, this particular time itself, your marks will be super high because of this particular series. Okay, so yes, rediscovery of his work happened in the year 1900. Okay, in the year 1900, his work was rediscovered by three main people, and you need to remember their name. Their name comes in the form of a question. One was Carl Correns, the second one was Eric von Schemark, and the third one was Hugo de Vries. Three people, Correns, Schemark, and de Vries. Correns, Schemark, and de Vries in the year 1900. Please remember, do not write 1990 and come, it is 1900. Okay. By this time, there were better microscopes, and it revealed chromosomes also. Chromosomes means what? Colored structures. So chromosomes were visible in the nucleus, and it was found that chromosomes basically they replicated and they split during the cell division process, and everything was super great. Okay, super great. It's Habiba. Hi dear, a very good afternoon to you. Okay, so this was being introduced. Rediscovery of Mendel work 1990, Correns, Schemark, and Hugo de Vries. Now, my dear children, 2 years after that, just 2 years after that, in 1902, two other heroes also came into picture. These were those two heroes, and those two heroes were none other than Walter Sutton and Theodore Boveri. Okay, Walter Sutton and Theodore Boveri. And they said that the behavior of chromosomes was parallel to the behavior of genes. And yes, so behavior of chromosomes was parallel to the behavior of genes means what? Just like how Mendel said that basically in a pair of alleles or pair of factor, when one allele is going to go into one gamete and another allele is going to go into what? Another gamete. This is, just a second. I think that side of the board it's not working. Yeah, he said that if it was capital T small t, one factor goes to one gamete, another factor goes to another gamete, right? So now these guys came into the picture and said, he told about factors, right? Basically, he might have been right because these factors are there on the chromosome. So when this chromosome goes to a gamete, whatever factors are there on these chromosomes or whatever genes are there on these chromosomes, that also goes. So that's when everything made sense. So Walter Sutton and Theodore Boveri said the behavior of chromosomes was parallel to the behavior of genes that Mendel studied. And that is how they gave down one very important thing, and that is chromosomal theory of inheritance. He said pairing and separation of pair of chromosomes would lead to segregation of a pair of factors they carried. Pairing and separation of a pair of chromosomes will also lead to pairing and separation of the characters or the pair of factors that they carried. This was chromosomal theory of inheritance. Did you get it? Yes, Subhash. Yes, we are coming to that. Guys, everyone, did you get it? Mendel had a downfall in 1900. Correns, Schemark, and Hugo de Vries said that no, he is actually correct. In 1900, they rediscovered their work. By then, you had better microscopes. 2 years after that, two people came into the picture. One was Sutton, and the other one was Boveri. And they said that pairing and separation of chromosome is parallel, is similar to what pairing and separation of factors. If it is clear, can you guys give me a thumbs up? If it is clear, give me a thumbs up. Then only I will go ahead. Come on. Yes, ma'am. Nagaraj, thank you so much, dear. Okay. Now, since that is clear, I have a question to you. This is from your PU2, and that means factors also segregate. Yes, dear. So when I say that a chromosome is there, okay, this is a chromosome. Whatever factors are there, factors are nothing but genes. Okay, on each chromosome you have genes, and these genes, my dear children, suppose I have a gene for height over here. In the same location, I will also have another gene for height. Now these genes, the different form of genes are known as alleles. Suppose I say one is capital T allele, one is small t alleles, but both of them are coding for a height only, no? Are coding for the same character, that is height. So these are alleles of a gene coding for height, and basically they are on the chromosome. So when the chromosomes separate, they are also going to segregate. Done? Clear? Ma'am, does it help to get out of out? Yes, dear, it will definitely help you. What others think? What others who are studying right now over here, do you think that you will get out from this particular series? You have been watching the video for almost 1.5 hours now. Yes? So your answer for this question is what? Which three scientists independently rediscovered Mendel's work? Subhash says the answer for this question is A. A. Nagaraj says it's D. It is what? D. It would be D. This is Dihybrid, Correns and Shemak. The answer for this question is D. Got it? Clear? Super clear? Yes, Winter Luna also gave me the correct answer. It's Habiba. Can you check the answer, dear? Can you check the answer once? Okay. Shall we go ahead with the next question then? Next question, or I think the next concept. So if this is super clear, let's go ahead with the next concept, which is going to be what? Which is going to be proof of chromosomal inheritance theory. So people gave chromosomal theory of inheritance, but you need to prove that as well. You cannot just believe it like that, no? You need to prove that. That is chromosomal theory of inheritance. And now came Thomas Hunt Morgan. Who came into the picture? Thomas Hunt Morgan. And basically you can see over here, who are they? These are not honeybees. They are basically, I wanted to put the picture of Drosophila. Okay, but yes. Okay, so listen, they are Drosophila. So Thomas Hunt Morgan, or T.H. Morgan, is a person who went ahead and tried to prove the chromosomal theory of inheritance by his experiment. And he worked on Drosophila melanogaster, which you also call as fruit fly, my dear children. But why did Thomas Hunt Morgan love fruit fly? Why didn't he choose anything else? Why fruit fly? Because there were few reasons, and this is your PU2 level question. Because they can grow easily on simple media. They do not need jalebi, aloo, kachori, anything. Okay, they do not need idli, dosa, nothing. They can grow on simple media. And also, they had a very short life cycle. If they have short life cycle means I can see any changes within them within 2 weeks itself. Within 2 weeks itself, you get it? So short life cycle is super important for me, who is going to do this experiment. Yes, they are also known as fruit flies, and they have high progeny count. If they mate once, they will produce so many progenies. I can study on these progenies. There was clear male-female distinction, and you know what? Males are looking smaller and females were the ones which looked larger. And visible hereditary variations were also clear under low power microscope. Under low power microscope, everything was clearly visible, as in their variations also. So these are the few features out there why he chose fruit flies or Drosophila melanogaster. And now I am going to dive into the exact experiment that he did, and also what did he find. Nagaraj, slides are beautiful. Thank you so much, dear. Okay. So basically, listen to me very carefully. So did he find something else? With this experiment, did he find something else? He was there to prove chromosomal theory of inheritance. He found linkage and recombination. And now this funda is super clear. Okay, I'll tell you, this funda is going to be super clear if at all you listen to me carefully. So everyone who is watching this video, okay, if you have joined now, if you have joined before, just listen. If at all you miss this, you might not understand a lot of things. So I am saying that there are 2 chromosomes. Okay, Now here I have capital A gene, here I have small a gene. Here I have capital B gene, here I have capital B gene. This is one, small b gene. And there is one more scenario. I have capital A and I have capital B here itself. I have small a and small b here. Okay, cool. Now listen to me. These genes which are there, they are far away from each other. I call them loosely linked genes. What do I call them? Loosely linked genes. Why? Because they are far away from each other. A and this B is very far from each other. But here they are very close to each other. I call them as tightly linked genes. They are very close to each other. I call them tightly linked genes. Okay. Now suppose I cut it over here like this and I make this portion to be interchanged. In both this condition, I make this portion to be interchanged. What is a new thing that I am going to find? I will see a chromosome over here with small a coming this side but capital B still over here. Capital A coming on the other side but small b over here. Do you find a new combination? Here it was capital A, capital B on this side, but here it is small a, capital B on this side. This means that a new combination is being made. A new combination of genes are being made. This is known as recombination. Recombination. Now, okay, here if at all you see, if this and this part is swapped, just a second. Yeah. What are you going to find? Small a, small b came this side, capital A, capital B went that side. Aren't these genes still together? Small a, small b is still here, capital A, capital B still there. So can I say recombination which is there, it is going to happen less? It is going to happen, why? Because the upper part is different. That is why recombination is there, but it is going to be less. On the comparison, in comparison to that scenario, my dear children, where the recombination is happening way too much. So loosely linked gene shows high amount of recombination. Tightly linked gene shows less amount of recombination. Did you get it? Something similar to euchromatin, heterochromatin. Yeah. That is little different. Yes. Did you get it? If you got it, just let me know. Yes, ma'am, I got it. Can you type it down? This is super important for you because I'm going to go ahead and explain you something. When the genes are loosely linked, okay, loosely linked, they show high recombination. It's very simple. You can remember like this. You have a friend. You have a friend and you think that basically, you have a friend, but you guys are not too close. When you guys are not too close, one more friend can also come in between. A new group can be formed. But if you and your best friend have a strong friendship, do you think anyone can come in between? No. That means less recombination out there. So if two genes very close, less recombination. If two genes very far, more recombination. Got it. Thank you so much. Now listen to me. So in his experiment, he didn't go by A, B, C, D and things like that, right? He did his experiment on fruit fly, and when he did the experiment, he got a chart like this. Now this is very complicated for us to understand, and do you think it's super important for us to know about this? No. It is important for us in exam perspective to know about this particular thing. And what does this particular paragraph say to you? This is super simple. Tightly linked genes very close on chromosome, very low recombination. Example, white and yellow genes. So there is a thing that is, okay. There is a thing that is a chromosome which is white eye color and yellow body color. White eye color and yellow body color, and then there is another thing which is what? Miniature wing. Wing size is what? Miniature. Now you need to understand this very important thing. I'll make it in bigger thing so that you get to see it properly. I'll make it over here. This is what? White eye, this is yellow body, and this is miniature wing gene. white eye gene is over here, yellow body gene is over here, and miniature wing gene is over here. Now, since these two genes are very close to each other, do you think they will show high amount of recombination? Do you think they will show high amount of recombination? They are very close to each other. High amount of recombination will be there? No. Less amount of recombination will be there. And that is exactly written over here. White and yellow genes show just 1.3% recombination. This means very less recombination. But what about white and miniature wing gene? This shows 37.2% recombination. What is the maximum recombination percentage? It is 50%. And out of that, if at all it is showing 37.2% recombination, that means what? It is far away from each other. So when he was studying about this, when he got to understand all this, he came up with the term linkage and recombination, my dear children. I hope this is super clear. Always remember white and yellow. White eye, yellow body, 1.3%. But wing is different. White and wing shape, which is there, they are far away from each other. More amount of recombination will be there. Done. This is the only thing that you need to remember. And with this, one more very important thing happened. That is, the student rocked, but the teacher got shocked. Who is teacher? Who is student over here? This is our teacher, that is T.H. Morgan, and he had a student whose name was Alfred Sturtevant. Alfred Sturtevant. What did he do so that he shocked the teacher? He did only one very important thing, and that is known as gene mapping. And that is known as gene mapping. What do you mean by gene mapping? So my dear children, if I know the percentage of recombination, I also know that they are far away from each other. If I know they are far from each other, depending on the percentage of recombination, can't I find their location? Yes, I can say that this gene is over here. This gene is far away from this. It is over here. That is why it shows this much amount of recombination. So the teacher found the recombination percentage, and with the help of that, the student find the location of the gene, and that is known as gene mapping. And if you have understood this this concept, then obviously, my dear children, so Alfred Sturtevant, I'm just talking about him, you can try to solve this particular question. Everyone, come on. If you understood the concept, I want you guys to solve this question. Understood, ma'am. Yes. Okay. In Morgan's experiment with Drosophila, when yellow-bodied, white-eyed female was crossed with brown-bodied, red-eyed male, and their F1 progeny was intercrossed, what was the percentage of recombinance in F2 generation? What was the percentage of recombination in F2 generation? That is the question. What is the percentage of recombination? Any guesses? Subha says the answer for this question is D. Make sure you read the question carefully. Read the question carefully. What are you crossing? Yellow-bodied and white-eyed female was crossed with brown-bodied and red-eyed male, and their F1 progeny were intercrossed. What was the percentage of recombinance in F2 generation? Those who understood the concept will be only able to answer this. Nagaraj said C. Nagaraj said C. So Nagaraj is the only one, and then Subhash is also saying C now. It is C, 1.3%. Why 1.3%? I told you, yellow-bodied, white-eyed female. White eye is there, yellow body is here, and whatever your other thing that you are crossing with, anyway, these genes are going to be very close to each other. Do you think you will find much recombination out there? Only 1.3% recombination is going to be there. If at all you're talking about miniature wing size, it is going to show what? If we are showing miniature wing size, it is going to show 37.2. Got it? The answer for this question is C. Yes. It's Habiba says the answer is C. Sorry, ma'am, it's C. Winter Luna. Okay. At least you got it. That's the whole purpose, right? So the answer is going to be C. Okay. Now this type of question, when it comes, we need to be more aware. Please go and revise it once. Also, one very important thing. Okay. Now, do I have any other question? Yes. Try to solve this question. Gene mapping technique or technology was developed by. Try to solve this one. Gene mapping technology was developed by Correns, Sturtevant, Mendel, Schemark. Correns, Sturtevant, Mendel or Schemark. gene mapping was developed by? This is an easy question, right? Very easy question. Two. Come on. I'm waiting for you to answer. B. Oh my God. Subhash B, Nagaraj B, and it's Habiba B. Winter Luna also says it's B. Okay. Since you're saying it's B, let's lock option B, that is Sturtevant. What was his name? Alfred Sturtevant. Okay. Alfred Sturtevant. He is a student of T.H. Morgan. Now, listen, there is a PU2 question that you got from the same concept, and that is recombinants obtained in both Mendelian and Morgan's experiment, yet the process involved are different. Comment. So in Mendel's experiment, when he saw the dihybrid cross, no, you saw that there was non-parental character. Remember, non-parental character, 9:3:3:1, three of them in between, they were non-parental. Why was it happening? Because of law of independent assortment. Okay, there the new non-parental character were coming because of law of independent assortment. But over here in Morgan's experiment, it was happening because of something which is known as crossing over. Okay, crossing over between the linked genes. When there is crossing over happening, that's when you found what you call recombination happening up there. So two things are separate things. Make sure you read this question really well and then only go ahead. This is an MQP1 question. Okay, in Mendel's process, recombination occurs due to independent assortment of genes on different chromosomes. All the genes were independently assorting. From capital R, small r, one guy was coming. From capital Y, small y, only one guy was coming. That's why. In Morgan's experiment, recombination occurs due to crossing over between the linked genes. Okay, done. Now, next thing. Differentiate between linkage and recombination. Again, this concept was again asked in MQP2. So this is something that is super important. You might get a question related on linkage and recombination in your boards as well. Got it? So linkage is what? It is a tendency of the genes located close together on the same chromosome to be inherited together. What is it? It is a tendency of the genes which are close together to be inherited together. Recombination is the result of it. It is a process by which new combination of genes are being formed after crossing over, after the exchange is happening. Crossing over is what you say. Now, it reduces the chance of new gene combination linkage. Yes, but whereas recombination, it increases the genetic variations out there. New combinations is going to be formed. Yes, physical association between what do you call the genes that is there, it is known as linkage. Cool. It's a 2-marker question. Now, do we have one more question? Yes. What are the conclusions drawn by T.H. Morgan's experiment, crossing over experiment? He said the higher the linkage, that means the tightly linked genes, the recombination is going to be less. The lesser the linkage, that means if they are far away from each other, more recombination will be there. In other words, genes which are tightly linked will show lesser recombination. The genes which are loosely linked will show higher recombination. Same thing, different questions. Okay. Now, T.H. Morgan selected fruit flies for his genetic experiments. Why? So whatever reasons we studied, you have to write all those reasons right over here. So I've given that. You'll get the PDF if you join our WhatsApp channel. So make sure you join the WhatsApp channel. Now, we are going to go ahead with the next thing, and that is concept number 8, sex determination. Are we ready? Are we ready for this concept? Question number 8. Let's go. Yes, ma'am. What about others? Again, guys, please drink some water. Yes, Habiba, you can get the PDF of the class. You just need to join our WhatsApp channel. Okay, just join the WhatsApp channel. The link will be given in the description session. Please join the WhatsApp channel and then we can proceed. Ma'am, 5 minutes break. Do you want 5 minutes break, everyone? So if everyone votes for 5 minutes, do you really want 5 minutes break? I can give if everyone gives me thumbs up. I don't mind. It's you guys who are important to me. Chai. Okay. Fine then. Yes, ma'am. Fine, fine, fine. So I'll take a 5-minute break. And you also take a 5-minute Kit Kat break. Okay, and if you're talking about 5 minutes, then you have to be back by, let's make it 6 minutes, 4 o'clock. Okay, be back by 4 o'clock. I'll be right here waiting for all of you guys. Go, 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 go. And when you go, when you come back with a lot of your friends, okay? Bye. Okay. So the break is officially ended. I'll just wait for everyone to come back. Hi, study. Ma'am, it's 4 o'clock. Yes. Hi, study. JST to you. Oh my God, big name. We are back. That's so good. So did you bring your friends also? Okay. So I'll just wait for everyone to come along. Okay. Just you guys are so punctual. Kudos to you. We'll just wait. Okay. So that everyone comes and then we will begin. Yes, Winter Luna is also there. Fine. Then shall we go ahead? Shall we begin? Nagaraj is missing. I think Nagaraj is on the way. Exactly. Back, ma'am. Okay. Nagaraj is also back. Since everyone is back and Subhash says example for incomplete dominance 4 o'clock planned. Exactly. Chalo fir, full Josh, we are going to go ahead with sex determination in the case of organisms, and it is a very fun topic. It's super easy, it's super fun. Let's see what's in there. So Baba, there are two types of things. One is what? Male heterogamety, and the other one that we have is what? Female heterogamety. Male heterogamety means males are going to produce two type of gametes, or males are going to produce two type of gametes. For example, I say human beings. Okay, in human beings, you might have seen we have XX chromosome for females and XY chromosome for males. Here, males are the ones that produce either X chromosomes or Y chromosomes. From this pair of chromosomes, only one guy can come in, either X or Y. But two type of gametes are produced by whom? It is the males. So this is an example of what? Male heterogamety. This is an example of male heterogamety. The same thing is seen in the case of Drosophila also. Drosophila also have XX, XY type of sex determination, just like human beings. Now, there is a twist. There is something else, something very, very new, and that is what? XO type of sex determination, and that is seen in the case of grasshoppers, my dear children. So in that, the females are going to be XX, just like how in human beings they are, and in the case of males, they are going to be XO in condition. They are going to be XO in condition. That means they can have either X chromosomes in a gamete, or they would not be having any chromosomes in the gamete. That is what you mean by O condition. Any sex chromosome would not be there. Okay. So anyway, they are producing two type of gametes, one with X chromosome, another with nothing out there. So that is why you say it is also an example of male heterogamety. So insects such as grasshopper show XX, XO type of sex determination, and this is example of male heterogamety. The same thing goes for what you call Drosophila as well as human beings. They also show the example of male heterogamety. Done? Clear? All clear? Now we have one thing, that is female heterogamety. So females producing two different type of gametes. Now this is very, very rare. This is seen only in the case of birds, my dear children. So in birds, what is so unusual? In birds, the males are going to be ZZ. And the females, my dear children, are going to be ZW. W for women, you can remember like that. So ZZ male, ZW female. Now when you form the gametes, the males are going to produce only one type of gametes. All of them contain Z only. But the female over here contains either Z or W in their gametes. Either Z or W in their gametes. Which is why this is an example of what? Female heterogamety. What is an example? Birds. Birds are the example for female heterogamety. Do not forget it. Super important question. Okay, in PU2, in KCET, both this examination, it is super important. Done? Clear? Now, what if I tell you there is one more type of sex determination, and that is going to blow away your head, and that sex determination is known as haplodiploid sex determination. What is it known as? Haplodiploid sex determination. Background sound, ma'am, somebody is teaching. Is it? Do you get background sound? No one is teaching. There is no one teaching. Reports clear? Now is it clear? Hi Narasimha. Hi dear. Okay. So over here in haplodiploid sex determination, just understand. Diploid means you have double the number of chromosomes, that is 2N condition. Haploid means half of it, that is N condition. Okay, half the number of chromosomes. So if I say that we have 46 number of chromosomes, our gametes usually have what? 23 number of chromosomes, that is half of it. Cool. Now over here, in this particular case, in the case of honeybees, the females are going to be diploid. For them, 2n is what? 32 number of chromosomes. Okay, 2n is what? 32 number of chromosomes. In the case of males, n is equals to what? 60 number of chromosome, that is half of it. So if it is haploid, it is going to be male. If it is going to be diploid, it is going to be a female. But how do you think that can actually happen? There has to be something, no? Why do you think that actually happens? How can diploid form this, that? Listen to me. If a female is diploid, it will undergo meiosis to form gametes. Gametes will have only half the number of chromosomes. So half 16, another gamete will be having 16, right? In this case, the male is already haploid. It cannot undergo meiosis. It will undergo mitosis to increase the cell number. The number of chromosomes remains to be the same, that is 16. Now 16 over here, 16 over here. Suppose both these gametes come together. 16 plus 16, I get 32. 32 is what? Diploid. Diploid is going to be what? Female. If it is diploid, female. Then ma'am, how it is male? How is male being produced? So listen, one gamete which has not underwent any fertilization. Okay, one gamete which has not underwent any fertilization, it will transform into the male one. This process is known as parthenogenesis. Yes, Nagraj, it is known as what? Parthenogenesis. Different from parthenocarpy. Parthenocarpy is seen in the case of plants. Okay, so parthenogenesis is there, where without fertilization you get what? Males. Got it? Is it clear? This is super important. Got it? So haploid-diploid sex determination is found in the case of honeybees. And while I say this, I also want to make you understand this very important thing. The males, do you think they have fathers? No, they do not have fathers. Do you think they have grandfathers? Arey bhai, this particular thing came from the female, from the mother. The mother had a father, that's how the mother is being formed, right? So it has a grandfather but it does not have a father. I know it's tricky, but it is super important. It does not have a father, but it does have a grandfather that is there. Okay, got it? And now we solve questions on the basis of this. Try to solve the first question. This is from your KCET. In male heterogametic type of sex determination, what is going to happen? In male heterogametic type of sex determination, what is going to happen? Female parent produces dissimilar gametes, male parent produces dissimilar gametes, male do not produce gametes only, and male parent produces similar gametes. Subhash, Baba, humans are not example of this. Humans are example of XXXY type of sex determination. Okay? Okay. B is the answer according to Nagraj. Nagraj says the answer is B. Winterfun also says B. Legend Soul Gaming is back saying B. Nagraj also says it's B. Okay, since you guys are saying B, let's log B. What does B say? Male parent produces dissimilar gametes, and that is what you mean by male heterogamety. You see in two type of organism. Sorry, you see in humans and Drosophila XXXY type of sex determination, and you see in insects XXXO type of sex determination. Both of their males are producing two type of gametes. Cool, you got it. You guys are awesome. Okay, now try to solve this question. XO type of sex determination and XY type of sex determination are the examples of what? Now you think these questions came in KCET. If at all you are giving exam after listening to this lecture, how many marks would you have scored from this chapter? Full marks. Full marks, no? So XO type of sex determination and XY type of sex determination are the examples of what? Subhash, can you just reload once? I think it's not stopped for anyone else, because even I am able to see clearly myself in my phone. So can you just once just go back from YouTube and come back and reload it? So answer is, Subhash says the answer is A for this question. Nagraj says it is B. It's Habiba says A. Bye. Read the question carefully. Legend Soul Gaming says it's B. Okay, Subhash says it's B. Winter Luna also says it's B. XO, XY type of determination, my dear children, it shows male heterogamety. Here males are producing two different type of gametes in the case of insects. Here again males are going to produce two different type of gametes in the case of human beings and Drosophila. So your answer is male heterogamety. Okay, now next question. From the chromosomal complements given below, identify the one which shows female heterogamety. Yes. Yes. Try to solve this. From the chromosomal complements given below, identify the one which shows female heterogamety. Now it's correct. Okay, cool. It's Habiba, now you were correct. Narasimha, video saved on YouTube. Yes, it will be there on YouTube. You can watch it later also if at all it's necessary. But I'll recommend you to watch it live. If you have doubts, you can ask us right now, right? Okay. Option D is Nagraj is what is saying. Legend says it's D. Now Habiba also says it's D. Okay, what is D? Okay, ZZZW. Winterluna also says it's D. Okay, everyone says D. Subhash also says D, which is why we log D. This is the only place where you see female heterogamety. This is seen in the case of birds. The males are going to produce same type of gametes. Females produce two different type of gametes. One is Z, one is W. W for women. You can remember like that as well. Cool? Chalo. Now, do you have another question? Yes. You have question from your PU2. So, PU2 question is, what is haplodiploid sex determination mechanism? Mention an animal which exhibits it. What is haplodiploid sex determination mechanism? It is where sex of an individual is determined by the number of chromosomes set it inherits. Males develop from unfertilized egg and are haploid. Females develop from fertilized egg and are diploid. That's the only thing that you need to write. And then this make this whole thing. And you get your marks. Okay? Okay. Now we are going to go ahead with the next concept, that is concept number 9, and which is mutation. Okay, which is mutation. Are we ready to go ahead with mutation? It's okay if at all we go ahead with mutation? I hope we can. Let's go ahead. So what exactly do you mean by mutation? Is mutation a good thing or a bad thing? I'll say it's both. It's both good and bad. Some mutations let new variations to come in and let us survive. Some mutation does not let us survive, makes us dead. Okay. So mutation is a phenomena that is going to alter your DNA sequence. And when your DNA sequence is going to be altered, obviously it is going to bring out changes in the genotype as well as phenotype, the physical appearance of how you guys look, right? Exactly. Thank you so much for giving me a thumbs up. All of you are super good. Lots and lots of love. Biggest hugs. If at all I have seen you real, I would have given you big hugs. Anyways, now coming to the examples. You have different types of mutation. One is point mutation. Okay, one is point mutation. Point mutation means change in a single base pair of the DNA. Okay, there is change happening only in a single base pair of DNA. For example, you might have heard about this disease, that is sickle cell anemia. Have you heard about sickle cell anemia? Over here, always remember, you have your DNA. The DNA transcribes into RNA, and whatever RNA is there, RNA is going to code for proteins. So in the RNA, you will be having codons. Okay, you will be having multiple codons. So there is a codon, suppose, and this codon is GAG. This is GAG codon. This is the 6th codon. You have 1, 2, 3, 4, 5. 6th codon is GAG. Now, because of some mutation, what has taken place is that one base pair has got changed. Instead of A, now it is replaced by U. Instead of A, it is now replaced by U. Now, GUG codon which is there, it is going to code for some another amino acid. GAG was coding for what? Glutamic acid. But GUG is going to code for some another amino acid which is known as valine. Now, if one amino acid is going to change, obviously the whole protein structure is going to be changed. So this is an example of point mutation where only a single base pair change had led to some problem out there. Always remember, example is sickle cell anemia. And I'll tell you easy way to remember it so that you never forget. Some people are like, was it GAG first and was it replaced by GUG? What is happening over here? Simple. A comes before U, right? A comes before U in the alphabetic order. So it was GAG first. It is replaced by GUG afterwards. U comes afterwards. Now the next thing, glutamic acid is the amino acid which is being coded over here. Glutamic acid. Now this particular codon codes for another amino acid, that is valine. So G comes before V. So initially it was glutamic acid, but now it is what? Valine. You remember like that, and then you will never ever forget it. Okay, clear? And now good examples. Yes, thank you. Now frame shift mutation. If at all you talk about frame shift mutation, it is caused by insertion or deletion of a base pair. For example, in your NCERT there is an example given, red cap. Red cap. Suppose these are the codons that we are having. So we read it as red and we read it as cap. Suppose I insert something else over here. I insert A over here. So now it will be what? Red and then ACA and then P. Don't you think the reading frame have shifted? Anyway, only three codons are being read at a time. So if at all you're inserting something, or basically if at all you're removing something, the whole reading frame is going to be changed. The whole reading frame will be changed. You need to understand that. Okay, that leads to what? Frame shift mutation. That is super important. Now suppose I am putting 3 of them all at once. Okay, 3 nucleotide all at once. So it was red cap initially. Now in between that I am putting something else. HAT. Now it is red hat cap. Whatever amino acid this is coding for, this is still code for that. This is also coding for amino acid. There is a new amino acid that is going to come up over here. Now always remember this very important thing, that is sometimes suppose the codon which is being added, it's a stop codon. It's a stop codon, then the whole process of translation is going to stop. It is not going to read ahead. So read the questions carefully if at all it's a stop codon, if at all it's not. Okay, if it's a stop codon, then always remember after red, if it is UAA, it is not going to be read ahead, it will be stopping it. Okay? Ma'am, when is our chem class? Chem class is going to happen tomorrow. Okay, so be there for the chem class as well. Fine. Now we will solve a chutku question on the basis of this. Try to solve this. Sickle cell anemia is due to the following mutant gene. Sickle cell anemia is due to the following mutant gene. What do you think is the answer for this question? This is a KCET question. Now this is kind of confusing question also. D, it's GAG to GUG. It is GAG to GUG. Okay. D. So in the answer key which was given there, the answer was A. In your KCET answer keys which was given there, the answer was A. But I would not say that you guys are wrong over here. Okay, I would not say you guys are wrong over here. But you need to understand one very important thing. That is, if I say the RNA has some change, sometimes the change might have come from the DNA itself. Whatever was there in the DNA, on the basis of it, the RNA was being made. Or the change can directly come in the RNA also. So suppose you're talking about mutant gene, okay, mutant gene, then over here, you're talking about the DNA. Suppose over here in the DNA, it was CTC first, but the T got replaced by A. The T got replaced by A, which is why over here, now when you make the RNA for it, it is going to be GUG, because you make the complementary RNA strand in transcription. You get it? So it was CTC, then it was GAG, but now the T got replaced by A, which is why now the RNA will be GUG. So there is a slight disparity in that, but still I will say. And here we are talking about mutant gene which is there. So gene are there. Gene is basically part of a DNA. We say it like that. But I get it. It can have this answer also to some extent. So this question is something that we can keep on hold. The reason is, okay, someone was saying, Winter Luna is saying, How, ma'am? So how is this only? So, listen, so RNA is this. When I say it is whatever is there in the DNA, opposite of it, complementary RNA strand is being formed on the basis of what is there in the DNA. Suppose the DNA has the mutation, then the RNA will also have the same thing. So it was CTC first, then the RNA strand will be G, A, G. But here if a T is replaced, my dear children, if it is replaced by A, then now it will be G, U, G. You get it? So it can come from your DNA as well. So that is why this question is like a bit confusion wala question. But yes, as of now you can keep it on hold. This is a KCET question, do not worry. We'll see what can be done. Probably it was not being questioned back in the year 2018 it came. And then the answer key it was mentioned like this. But I also totally understand that the answer would be D itself. Okay? So this type of questions may come. Have your insights very, very clear. If at all you see a question is too confusing for you guys, attempt it anyways. You will get marks if the answer keys are wrong. Both are marks, if at all, if it is marked in your KCET examination, it is not going to be considered. Only one can be accepted. Okay, anyways. Define point mutation, give an example for point mutation. This is your PU2 level question. Change in single base pair of DNA is known as point mutation. And a classic example is of sickle cell anemia. Just explain that, that's all. Done? Done. Now we go ahead with the next concept. We have 3 more concepts. Someone was asking me how many concepts are left. 3 more concepts are left. Okay. So concept number 10, Mendelian disorders, and this is where everything is going to go super, fun and energetic and nice. Okay, it is going to be really, really nice from this point onwards. You'll have a fun class from here. So what exactly do you mean by Mendelian disorders and what are chromosomal disorders? So we have 2 genetic disorders, one is Mendelian and this chromosomal. Mendelian means, suppose over here I say that there is a chromosome, and in this chromosome there is a gene. Now I am saying this gene is getting mutated. Some changes are happening in this particular gene, which is why whatever X particular protein had to be made is not being made. This is how Mendelian disorders are being caused, because of the gene's mutations or some problems around in this particular gene, determined by alteration or mutation in a single gene out there. So whatever function it was doing, it won't be doing now. Chromosomal disorders means, for example, I say we generally have a pair of chromosome. We generally have a pair of chromosome each. Suppose I'm adding one more chromosome over here. Baba, there will be more amount of synthesis being done. There will be more problems. Or suppose instead of having 2, we just have only 1. This is a problem because you need 2 chromosomes anyway. So chromosomal disorders arise when there is a problem with the number of chromosomes or the structure of the chromosome. That is the thing. And the first topic that we are going to discuss is Mendelian disorders, wherever a gene is mutated and because of which we will be having diseases. And the first one is color blindness. The first one is what? Color blindness. This is a sex-linked recessive disorder. That means the genes which are there, they are there on the sex chromosome. So before I go ahead, I think I have to talk about one more very important thing, I'm sorry, and that is, in our body, how many chromosomes are there? 46 number of chromosomes. So these 46 number of chromosomes comes in 23 pairs. Okay, 23 pairs. 22 of the pairs, my dear children, you call them as autosomes. They are generally there, they are responsible for all our physical character and everything. The last one, the 23rd pair that we have, they are known as sex chromosomes, and they are also known as allosomes. They are known as sex chromosomes and they are also known as allosomes. Okay, done. 22 autosomes, 23rd sex chromosome or allosome. If at all in this particular chromosome there is a gene and that gene is getting mutated and that is why we get diseases, this will be known as sex-linked disorders. Okay, this will be known as sex-linked disorders. So the first Mendelian disorder is the sex-linked recessive disorder, which is color blindness. Now people often think color blind people cannot see any color. Is it like that? No, Baba. They can see colors, but they won't be able to see selective colors out there. So over here, the most common type of color blindness is where people cannot see red and green cells, green color. Why? Because they have some defect in the red and green cone cells of their eyes. And it occurs in about 8% of males and only 0.4% of females. Why do you think that is happening? Why this partiality over here for the males? Why 8% of males are affected and only 0.4% of females are affected? I am going to talk about that, but please remember this number, 8% males and 0.4% females. Okay, listen to me carefully, how this disease is usually being passed on. So, okay, the first situation is where the male is normal, XY is for a normal male. The female, my dear children, is a carrier. Is what? A carrier. That means only one gene is being affected. She has another gene, is X chromosome. X chromosome will be having another gene. Everything can be controlled by the other set of gene which is there. So it's totally fine. She is just a carrier and he is a normal guy out there. When this condition happened, let's see what is going to be the type in progenies. When this and this fuses, you get a normal female. When this and this fuses, you get a carrier female. Why carrier? Because one X is she's carrying one mutated form of X gene, and rest she's not diseased. Why? Because she has a normal X chromosome also. When this and this comes together, it leads to a condition like this, and I'll tell you what exactly happens. And when, sorry, just a second, okay? I think I did a mistake. Okay, when this and this comes together, I get a normal guy, and when this and this comes together, I get a man who is colorblind. Ma'am, how is that fair? Here also only one chromosome is being affected. Here also one chromosome was being affected. But Baba, do you think it has another X chromosome to overshadow the effect? No, there is no other X chromosome to overshadow the effect, which is why this guy will be what? Colorblind. This guy is going to be normal. This girl is also going to be normal. This girl is also going to be normal, but she is what? A carrier. She's just going to be a carrier. Okay, got it? Now, if at all we go ahead and see the second scenario. Let's see the second scenario. The second scenario is when, went down. This was the first scenario. Now we might need space. So second scenario is when the male is again normal. The female, my dear children, she is colorblind. She is colorblind. Let's see what is going to happen over here. This and this comes together. It is XXXC. This is a normal girl, but she is what? A carrier. Normal female, but she is a carrier. This and this comes together again. She is going to be normal, but she is a carrier. Fine, she is not diseased, she is a carrier. That is what I mean. Now this and this comes together. XCY. He is going to be what? Colorblind. He is going to be colorblind. What about this and this? Again, Baba, he is going to be colorblind. If at all you see, the males are the one which are usually affected, and it is because of this particular reason. You want more examples? I can give you more example. Suppose, now if I want the female to be colorblind, the colorblind gene should be there on the father. So suppose the father is colorblind, my dear children, and the female is normal. Ulta. The father is colorblind and the female is normal. If I cross this with this, I get carrier female, normal, carrier female. This and this, I get what? Again, normal carrier female. This and this, normal guy. This and this, again, normal guy. So here also, the female is not being affected. The female is not being affected at all. So totally fine. That is also totally fine. Now again, if I actually want the female to be affected, I need to understand this very important thing. The female should be like this. That means one gene should come from the father, another gene should come from the mother. That means father should be colorblind. Because if father has one gene, that means he is colorblind. And the mother, my dear children, at least she should be what? A carrier. At least she should be carrier. So if now condition is like this, in this condition, when this and this comes together, that's when a female is finally going to be colorblind. With so many things being done, what did you understand it? With so many things being done, you understood one very important thing, that is 8% of males are being affected and only 0.4% of females are going to be affected. Females are usually carriers unless they inherit 2 defective copies, one from each of their parent, just how I showed you. Probability, a carrier mother has a 50% chance of passing it to her son. A carrier mother, so for example, we did an experiment over here. This is the carrier mother. She has a 50, out of the 2 sons out there, only 50% of the son got it. Out of the 2 sons, only half of the son actually got diseased. You get it? So basically the question is, the thing says, a carrier mother has a 50% chance of passing it to her son. A daughter will be colorblind only if her father is affected and mother is at least a carrier. At least a carrier. Okay. Got it? Narasimha, yes, we will be doing lots of questions on the basis of it. So children, I hope the concept is clear. Okay, and always remember, almost the same type of concept is there for hemophilia also. Hemophilia is also a sex-linked recessive disorder. That means why it is known as recessive disorder? If both the chromosome comes in recessive alleles, okay, for example, the females, they are like this, then only the female will be diseased. In the case of males, they do not have anyone to dominate. That's why, again, you call it recessive condition only. That's why the person will be diseased. Now, if we solve some questions, let's go with this. Okay, now here, my dear children, you have to make sure you use your brain a lot. A colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be what? The colorblind man marries a carrier woman. The percentage of their colorblind progeny in the next generation will be what? Come on, solve it. Subhash, you can ask me towards the end, okay? I'll answer it. Chalo, 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 chalo. A colorblind man. Colorblind man means what? He is going to be like this. Marries what? 50% is what Narasimha is saying, okay? Winterluna says B, okay? Winterluna in full form, okay? What about others? Others who watch the video? Carrier female means what? Carrier female is going to be like this. Only one is going to be affected. Only one allele is going to be affected. Nagaraj says the answer is B. Now when you cross this and this is going to be a carrier female. This is going to be an affected female. This is going to be a diseased female, a colorblind female. Now this and this, XY, that means normal person. This and this. Again, my dear children, if at all you see, this person is also diseased. This is a man who is diseased, this is a female who is diseased. So 50% out of 4 people, 2 people are affected. That means your answer will be what? 50%. You guys are super good. Okay, you guys are super good. Ma'am, which question, Jagu, okay, which question will be asked in HOTS? These type of questions can be asked in HOTS. Okay. So yes, your answer was B. You guys understood. Awesome. You guys are awesome if at all you understood this. Now we go dig deeper. Okay, let's try other question on the same concept. Both male and female have normal vision, though their fathers were colorblind and mothers did not have any gene for colorblindness. The probability of their daughter becoming colorblind is what? Both male and female have normal vision. Agreed. Normal vision. Okay, and their fathers were colorblind and mothers did not have any gene for colorblindness. What do you think is the answer for this question? Remember, if you're saying that the male is having normal vision, that means the male will be like this only. He cannot be anyway carrier. Okay, and female, my dear children, if you're saying she has a normal vision, but you are saying her father was colorblind, then obviously she can be a carrier. She can be a carrier. Carriers can also be having what? A normal vision. Carriers can have a normal vision, right? And father, if at all he was colorblind. If father is colorblind, obviously X gene comes from the father only. in girl child, one X gene comes from the mother, one X gene comes from the father. So anyway, this particular female might be colorblind if she needs to have a normal vision. Now you are asked one very important question, and that is the probability of their daughter becoming colorblind is what? 0, yeah. 0%. A Subhash is saying A, Jagannath is also saying A. Okay. Only you guys are answering. Let me see. In between, Nagaraj also said it's A, is it? Guys. And it's what do you call? It's Habiba also gave me the answer B for the previous question. Good. Answer is A, is what everyone is saying. Is it A? Let me see. So suppose this and this is being fused. When I cross this with this, I get XX, normal female. When I cross this with this, I get a carrier female, but again she is normal. This with this, I get XY, a normal male. This with this, I get a diseased man. But the question is the probability of their daughter becoming colorblind. So none of the daughters are colorblind. One is carrier, one is normal. So answer is 0%. You all were correct. It's Habiba also gave me the answer for this question. Awesome. Awesome, children. Superb. Now going ahead with the next question out there, and it goes like this. This is a PU2 related question. Try to solve this. A woman with normal vision whose brother is colorblind marries a man with normal vision. Okay, a woman with normal vision whose brother is colorblind marries a man with normal vision. Okay, they have 4 children, 2 sons and 2 daughters. One son is colorblind while the other son and both daughters have normal vision. The pattern of inheritance for the disease is what? Not Jagannath ma'am, Jagu. Okay, Jagu, fine. Do I have to call it twice? Jagu? Okay. What is the answer for this particular question? This is a very easy question. Here everything is given. A woman is normal vision, having normal vision, brother is colorblind, marries a man with normal vision. They have 4 children, 2 are daughters, 2 are sons, 1 is colorblind. So color blindness is the disease that you're talking about over here, right? And color blindness, we all know it is what? It is going to be what? Sex-linked recessive disorder. It is going to be sex-linked recessive disorder. Okay, one time. Okay. Now, Emma dear, I think it's just not clear for you because everyone else is answering. Can you try to zoom it in your screen? Yeah? Or if you want, I can just zoom it and show to you like this. But there is a problem. I cannot do multiple tabs. The woman, guys. So I think in your screen, if at all you zoom in, it will be more easy. Yeah? Chalo. So we did that. Now the second sex-linked recessive disorder, we are going to go get that, and that is hemophilia. And you know what? Queen Victoria had hemophilia. What is hemophilia? Again, it's a recessive disorder, sex-linked recessive disorder. Here, a single protein that is a part of a group of other proteins which is involved in clotting of blood is being affected. So basically, if I say that you need A, B, C protein for clotting of blood, but A is not being produced. Why? Because the gene for producing A is being mutated, and this gene is on the sex chromosome. That is why clotting is not going to happen. So basically, if at all I get a cut over here, it is going to go, blood is going to go like this. Now again, this is sex-linked recessive disorder following the same pattern like color blindness. But over here, you need to know one more very important thing. The possibility of a female becoming a hemophilic is extremely rare because the mother of such female has to be at least a carrier and the father should be hemophilic. If I say, no, color blindness is something that you can live with. Color blindness is a disease that you can live with, obviously. But if someone has hemophilia, do you think they can live with it? No, they cannot live with it. They will be obviously passing away after some point of time. It's not the same scenario now. Okay, but it was like that before. So basically, if I say that the female, the mother who is there, she is basically, where is that? Okay, female, suppose she is a carrier, and the male who is there, he is hemophilic. The male will be XY, and the male is hemophilic in nature. In that condition only, a female can get the disease, but you need to understand one very important thing. Do you think the male will be, alive until the time that he reproduces? The answer is no. The answer is no. Okay. No one, actually the female or the male, anyone who has hemophilia, they would not be alive until that particular time, which is why I said the possibility of a female having this particular disease is very, very less. Is going to be very, very less, because the male would not survive until the reproducible age out there. So that is one very important thing, and I hope that's clear about hemophilia. Done? Clear about hemophilia? Shall we move ahead? Yes, Jagu, got it. Now hemophilia are Saheli. Okay, oh my god. Why father won't be unviable? Unviable as in, because as I said, they won't be alive until that particular age. Listen, females have an option to be a carrier. Not an option, I'm saying. Females over here are a carrier at least. They can alive because they can be alive because there is one extra X chromosome supporting all their functions out there. The blood can somehow clot. Okay, she's just a carrier. But over here, the blood is not clotting. They won't be, how they will be alive if at all there is a cut happening in their body. That is very this is a normal thing, right? So, do you think they would be surviving? No, that is the reason. Ma'am, sorry, but can you sing a song? Jagu, if I sing a song in the examination time, you guys also would have to just sing a song, seeing the paper. Okay, because there won't be anything to write. Okay, chalo. Jokes apart, let's go ahead. I hope it's clear. Someone was asking. Yeah, Winter Luna, I hope it was clear. Yeah? Okay, ma'am, understood. Chalo. Now, the sex-linked recessive disorders have ended up over here, and I have a question for you. The gene for hemophilia is located on the X chromosome. Hence, it is normally impossible for what? Now, I told you, each and every line of your NCERT is important because you get questions like this. The gene for hemophilia, blood clotting disorder, not clothing. Why do you clothe a blood? It is clotting, C-L-O-T-T-I-N-G, clotting. Okay? The gene for hemophilia is located on dash chromosome. Hence, it is normally impossible for what? Come on. A. Nagraj says A. Hemophilic father to pass the gene to his daughter. Is that the answer? Option A. Hemophilic father to pass the gene to his daughter. That is something that is normally impossible. Exactly, yes. This is the answer because we just discussed it. Hemophilic father might not be alive until then to basically reproduce and pass it to the daughter out there. So Winter Luna, Nagaraj, both of you are absolutely correct when you said this. Now we go to the next question, next concept, not next concept, next disease that, just a second, this is marked wrong, okay? This is marked wrong, the answer is this only. Okay. The next is autosome-linked recessive disorder. Now this is again super important. There are three recessive disorders that you need to know, and they are all autosome-linked. Autosome means the rest of the chromosome. Apart from the sex chromosomes, the 22 chromosomes that we have are what? Autosomes out there. If at all there are some genes which are located on the autosomes and they are not working well, there is some mutation in them, it causes disorder. And the first example that we have is of sickle cell anemia. Okay, first one is that of sickle cell anemia. What exactly is happening in sickle cell anemia? So my dear children, before I go ahead with that, I need you guys to know one very important thing, and that is your RBC which is there. It is usually discoid in shape. Okay, there's a disc-like thing that is being formed. It is discoid in shape, and basically this RBC is going to carry a very important thing that is known as hemoglobin. Okay, hemoglobin is something that is going to carry what? Oxygen out there. Because hemoglobin is made up of two chains, alpha globin chain, and the other one is the beta globin chain. Both of them together forms the hemoglobin. Now suppose the beta globin chain which is there, it is not being made properly. That what is going to happen? What is going to happen? It is going to cause trouble. Yes, discoid is also known as bioconcave. Okay, it is also known as bioconcave shape as well. Fine. If beta globin chain or alpha globin chain has been affected, there will be problem in the hemoglobin. So this is exactly what is going to happen in sickle cell anemia. The beta globin chain is not going to be produced well. Why? Because over here I was talking about the 6th codon. It was GAG, but now it is replaced by GUG. That means it was initially coding for glutamic acid, but now it is coding for valine. And when it happens, one amino acid is going to change in the structure of the whole beta globin chain. One amino acid is now going to change, which is why the structure of the beta globin chain is being affected. If beta globin chain is being affected, hemoglobin is being affected. If hemoglobin is being affected, RBC is going to be affected. It cannot carry enough amount of oxygen. And which is why I'll say it will change its shape to what? Sickle cell. It will change its shape to sickle cell. And if it is changing in shape to sickle cell, what is going to happen? It cannot move around easily. It can form clots in our body. It can even form clots in our body. So oxygen transportation is less. It is forming clots in our body as well. That is a major problem in sickle cell anemia. There is a major problem that you see in sickle cell anemia. It is an autosomal linked recessive disorder. It's not dominant, it's recessive disorder. That means, suppose a person has a normal HbA gene, like this, it's all normal. If suppose a person has one HB gene being affected, still it will be normal, because this particular condition is a recessive condition. Both of the HB genes should be affected. That's when I say that a person will actually have a disease. So this is a carrier for sickle cell anemia. This is the person who is diseased in sickle cell anemia. I hope you got it. And this is very much common. And I'm telling you, okay, a friend of mine, her brother died because of sickle cell anemia. Because, my dear children, both of their parents were carriers. Now here males and females both can be carriers, and both of them, if at all they are carriers, just imagine, if both male and females are carriers, what is going to happen? Suppose this and this comes together. Don't you think that person will be having sickle cell anemia? Yes. So you and me might seem very normal, but we might be a carrier for some genetic traits, and we can pass it to the next generation out there. We never know. That is why we say that before planning for any children out there, we should be going and getting consulted with a doctor, because we might be carriers of diseases. Okay? Got it? So this is the thing. And sickle cell anemia, and that is the reason why initially, you know, long before, people used to marry between what relatives out there. Then it was said that you should not marry the relatives. Why? Because it is genetic. It is running in your blood. Okay, you get it? The same genes are being transferred. Now there are high chances that you and other person can be a carrier, and obviously the kid will also be diseased in this particular condition. Got it? Clear? Fine. I hope this was insightful, interesting, as well as scary. Okay, so beta globin gene, okay, the gene which is responsible for producing the beta globin chain, it is on chromosome number 11. Chromosome number 11. And over here, it's not the quantity of the hemoglobin that is being problematic, it is the quality. It is the quality that is being compromised, which is why you call this as a qualitative disorder. What do you call this? Qualitative disorder. Got it? Yes, please take a screenshot of this. You might not get it, because this is a new slide that I have added up. So over here, there's a question for you guys. You do not have to tell me the answer now. Don't tell me the answer now. People with sickle cell anemia won't have malaria. Tell me the answer in the comment section. In the comment section, not here in the chat box, in the comment section later on. So I'll read out the comment, I'll reply to you, I'll let you know. So yes, caused by mutation in the beta globin gene, glutamic acid is replaced by valine at the 6th position due to this substitution. Mutant hemoglobin molecule undergoes polymerization under low oxygen tension. It undergoes polymerization under low oxygen tension, changes in the change of the RBC from bioconcave disc to elongated sickle-like structure, reducing the oxygen transport and also causing blood flow obstruction. HbS homozygous condition is where someone will be diseased. HbS heterozygous condition is where someone is a carrier. Okay, and HbA is a normal beta globin chain. If both the parents are carrier, the child will definitely have a chance. It's not necessary, will have a chance to have it. if both of them are carriers. Got it? Ma'am, when does the session end? Soon, dear. Soon. Okay, so I hope that is super clear. Okay. Now, since anyway we're talking about sickle cell anemia, I will generally talk about one more very important thing that is related to sickle cell anemia, a blood disorder again, and that is thalassaemia. And Amitabh Bachchan is also a thalassaemia carrier. What exactly is thalassaemia? Listen, over here also again alpha chain, beta globin chain, super important for formation of hemoglobin is there, right? Now listen to me carefully. If you need to make alpha chain, okay, if you need to make alpha chain, you need to understand that there are in total 2 genes responsible for it. Okay, 2 genes. One gene having 2 alleles, another gene also having 2 alleles. Now over here, suppose some mutation is happening in this particular gene. Still alpha globin chain is going to be produced, but not much. Little bit alpha globin chain is going to be produced. If this is also getting mutated, again still it is going to be produced, but in less amount. If suppose this is also going to be mutated, again it is going to be less. If this also, then it is not going to be there at all. Alpha globin chain is not going to be produced. If alpha globin chain is not being produced, do you think hemoglobin can be formed? No, Baba. We say it's a major thalassaemia patient. It's a major thalassaemia patient. If at all only a few of them are being, only a few of them are damaged and other things are there, you say it's a minor case out there. Now, over here, understand it's according to the quantity of the protein being produced. It's not qualitative, it's quantity of this particular thing which is being produced, alpha globin chain or beta globin chain. So in alpha globin chain, 2 genes, 2 alleles for each of them. Right. Now in beta chain, you know it is on chromosome number 11, you just have 1 gene, 2 alleles. If this is affected, no beta globin chain, less beta globin chain. If both of them are affected, no beta globin chain is going to be produced. So this is going to be what? A quantitative disorder. Again, this is on chromosome number 11, and in the case of HbA, that is alpha globin chain, it is on chromosome number 16. Number of chromosome is also super important. Now thalassaemia is what? A mutation or deletion raises synthesis of either alpha or beta globin chain of hemoglobin, leading to what? Abnormal hemoglobin and also anemia. Alpha thalassaemia affects the alpha globin production in mutations in HbA1 or HbA2 on chromosome number 16 causes this particular disease. Similarly, beta thalassaemia affects the beta globin production in the HBB gene on chromosome number 11. That's all. So it's a quantitative disorder and sickle cell anemia was a qualitative disorder. Then, ma'am, go fast. If I go fast, you will not understand anything. Now, there is one more autosome linked recessive disorder, and that is phenylketonuria. This is what? Phenylketonuria. We already discussed about it. This is why I don't think I should discuss it. Phenylalanine hydroxylase enzyme is not being produced, which is why the conversion does not take place, which is why it is getting stored in the form of phenylpyruvic acid. And also, one very important thing, there will be reduced skin as well as hair pigmentation. Now, this compounds are excreted in the urine due to poor kidney absorption. This is also one very important thing. People who are having phenylketonuria, their urine smells so bad. Why? Because even the which is the phenylpyruvic acid which is there, it has not been absorbed back by the kidney. It is excreted through the urine and smells really bad. Cool. So the autosome linked recessive disorders are over. What are they? 3 of them. Please remember: sickle cell anemia, thalassaemia and phenylketonuria. One more is there, that is cystic fibrosis. Cystic fibrosis. This is written in the text format in your NCERT. This is also important. It is what? Cystic fibrosis. Please remember, it is an autosomal linked recessive disorder. And in your NCERT, every disorders which are there, they are all recessive. They are all recessive except one. Except one. Rest, everything is recessive. Okay, do you know what is that one? Do you have any idea what is that one? We'll solve the question and we will get to identify that also. Okay, by then, until then, try to solve this question. Identify the odd one among the following disorders. Hemophilia, sickle cell anemia, phenylketonuria, thalassaemia. Which one is the odd one among the following disorders? Hemophilia, sickle cell anemia, phenylketonuria, thalassaemia. Come on, come on, come on. Come on, children, come on, fast. The odd one. The odd one out of them, hemophilia is what? A sex-linked disorder, whereas if at all you see this, sickle cell anemia, phenylketonuria, thalassaemia, all of them, my dear children, are going to be autosomal-linked recessive disorders. So your only answer for this question is A. Exactly, Winter Luna. Exactly. Awesome. Let's go ahead with the next question then. What is the next question? Next question says, example for non-Mendelian disorders. Thalassaemia, hemophilia, cystic fibrosis and Down syndrome. Which is a non-Mendelian disorder? Mendelian disorders are the things that we studied until now. If you are asked about non-Mendelian disorders, is something that is not, what do you call, linked with this. Okay, whatever we studied. So the exception that we have not studied until now will be your answer for this particular question. What is the thing? Any guesses? What is your answer for this particular question? It will be Down syndrome. Thalassaemia, hemophilia, cystic fibrosis, all of them, my dear children, are Mendelian disorders. The only example is going to be what? Down syndrome. So your answer for this question is D. Is D. Yes, Therin, hi dear, and Subhash. Okay, going with the next question out there. Do we have a next question? Yes. If both parents are unaffected carriers for thalassaemia, which is an autosomal recessive disorder, what are the chances of pregnancy resulting in an affected child? What are the chances of pregnancy resulting in an affected child? Hi Saida. Okay, Saida also gave me the correct answer. If both of them are unaffected carriers of thalassaemia, unaffected carriers of thalassaemia. So basically a normal gene is there, and if they have to be carrier, they need to have the carrier gene also. So HBA, then HBT. This is carrier. Now you are asked, what are chances of pregnancy resulting in affected child? Let's see. If this and this comes together, you get HBA. This is normal child. Okay. If this and this comes together, you get HBA, HBT. That means you are talking about a carrier. If this and this comes together, you say HBA, HBT. Again, this is going to be a carrier. If this and this comes together, you get HBT, HBT, which is going to be disease. So out of 4, 1 is going to be diseased. That is means out of 4, 1 is going to be diseased. That means what? 1 by 4 is equals to what? 1 by 4 is equals to what? See ya. Winter Luna, check madi, then only you will be able to answer this. Subhash also see 1 by 4 is equals to be 25% are going to be affected in this particular case if both of them are carriers, right? Children, are you solving some other question? Unaffected carriers means they are carriers, they do not have the disease. That is the meaning of this particular question. In that particular case, when you cross, your answer is going to be this. It's going to be 25%. The answer is A, not C. It is going to be A. Okay? I hope you were solving the same question. Now, one more question is there. Okay. Now, you have to answer it fast, okay? Sex-linked recessive disorder occurring in about 8% of males. This is an MQP question, that is model question paper question. Sex-linked recessive disorder occurring in about 8% of males and only about 0.4% of females, leading to a defect in the cones of the eye. Basically, it is going to be color blindness. It is going to be what? Color blindness. No, ma'am, confused. Okay. It's okay. It's okay. It happens. It is going to be color blindness. Chalo. Next, sex-linked recessive disorder affecting a single protein involved in the clotting of the blood. What is B? Can you guys tell me what is B? If clotting of the blood is being affected and it's a sex-linked recessive disorder, what do you think is the answer for this particular question? B, 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 B. Come on. Hemophilia? Exactly. It is going to be hemophilia. Okay. Good, Subhash. Great, great, great. Next, C1. Inborn error of metabolism. That means it is there from the birth. Autosomal recessive trait. Accumulation of phenylalanine happens over here. Accumulation of phenylalanine means this disease is what? This disease is going to be, what is C? Yes, Saida. Great, great, great. What is C? PKU, that is phenylketonuria. The answer for this question is going to be phenylketonuria. Great. Sayada, Winter, Luna, all of you. Super, super. Now, autosomal linked recessive blood disorder that leads to substitution or amino acid in the beta globin chain of hemoglobin. Yes. What is the answer for D option? It is going to be sickle cell anemia. Because here clearly substitution is being mentioned. Okay. And the E1, the last one that we have, autosomal linked recessive blood disorder, reduced rate of synthesis of globin chains of hemoglobin. Your answer is going to be thalassaemia. Reduced rate of hemoglobin means thalassaemia. Always remember, in your NCERT there are 3 blood disorders. Hemophilia is also a blood disorder, but only 2 of them are going to be autosomal. One is sickle cell anemia, other one is thalassaemia. Hemophilia is sex-linked blood disorder. Cool. Now this came in your MQP. I have written the answer also. You will get it in the PDF. Please join the WhatsApp channel. Now few autosomal linked blood disorders in human population. Among them, some are related to qualitative and some are related to quantitative problems of synthesizing blood proteins. Explain. So which one is qualitative? Which one is quantitative? You should know. Qualitative is what? It is sickle cell anemia, and quantitative is what? Thalassaemia. And you have to explain about that. It's a 5-marker question, so don't miss it. I have explained it already, which is why I'm not explaining it again. Okay, but you will have the notes which you can refer later on. Similarly, explain thalassaemia as an example of Mendelian disorder. This also came as a question. MQP2 of this year. Make sure you practice all the questions similarly. Okay? Subhash, break. No, please don't ask for break. I'm going to complete the chapter with the next few minutes. Like maximum it can go is 15 minutes. Okay, so please don't ask for me break for me. I'll just complete it, and then you guys are free now. Then you guys are free. You can do whatever you want. Okay? Fine. So please explain that. And now concept number 11, the second last one, that is pedigree chart. That is what? Pedigree chart. So pedigree chart, my dear children, you need to know all this thing, all the things. Square is there, then it is male. Circle is there, then it's a female. If the shape is like this, that means sex is unspecified. The sex is not being specified. If all of them, my dear children, are colored, then it is affected individuals. That means they have the disease. If a square is there, one line is there, and O is there, that means mating is, circle is there, that means mating is happening between male and female. If, my dear child, over here you have 2 lines, now this is the most important one. If you have 2 lines, it shows mating between relatives, or it is also known as consanguineous mating. It shows mating between relatives or consanguineous matings. Now, over here, male and female came together, you got a daughter and a son, but none of them are affected. That means it's totally fine. Parents are mating, and then over here there are children being formed. But here male and female mated, but the child which is there, it had got a disease. It had got a disease. Okay, and if at all you have an icon being made like this, and over here you're writing 5, that means 5 children are being formed. The sex is not being specified out there. If it is colored, that means 5 of them who are there, the sex is not specified, but they have a disease. So what is pedigree? Analysis of traits in several of generation of a family is called pedigree analysis. You do it in several generations. Okay. Now over here, in your NCERT, the only dominant disorder which is given under this picture, and that is myotonic dystrophy. Do not forget this. It is autosomal dominant disorder. Now, how do you understand between a dominant disorder graph and a recessive graph? It's super simple. If at all the disease is found in every single generation, you say that this is going to be a dominant disorder. For example, here I see the mother is affected, here the son is affected in the next generation, and also the daughter. Here in their next generation, the daughter is also affected. In every single generation, don't see like this. See generation to generation. If this generation, this is the next generation is going to be this. In every generation, if at all someone is diseased, it is going to be a dominant disorder. On the other hand, if it is not there in every generation, if there is skipping of generation, in this generation it is not there, but in this generation it is there. In this generation it is not there, but in this generation it is there. This is going to be what? Recessive disorder. Okay, this is going to be recessive disorder. Got it? Got it? Fine. So myotonic dystrophy is an autosomal dominant trait, remember, and always remember, if at all you're asked whether it's sex-linked or autosomal, how do you find that out? Autosomal is basically when the males and females might be equally affected. It's not like the males are affected more. Usually in sex-linked disorders, the males are the one which is affected more, and in sex-linked disorder, you can see a crisscross pattern. Crisscross pattern as in from the father to the daughter to the son. Male, female, male again. So crisscross pattern can be observed in the case of sex-linked disorders pedigree chart. This is super important. If at all you know it, you will be able to solve any question out there. Now I have few questions. Try this, everyone. The affected male in the pedigree chart is symbolized by. The affected male in the pedigree chart is symbolized by. How do you show that a male is being affected? How do you show that a male is being affected? Come on, come on, come on, come on. A. Winterluna says A. Yes, it is A. A square and the color out there, that means it is going to be male who are going to be affected. The next one that we have. In the following symbols used in the pedigree analysis, identify the symbol that denotes consanguineous mating. That denotes consanguineous mating. What do you think is the answer for this particular question? Hi Rohan, Ruhi is also there. Hi Ruhi, says the answer for that question was A, correct. Now here if you want to show consanguineous mating, what do you think is the answer for this particular question? Consanguineous mating means mating between relatives. Mating between relatives. So in that particular case, what is going to be your answer? A, B, C, D. B. Winterluna says the answer for this question is B. Yes, the answer for this question is actually B. You can see two lines between male and female. That means it is going to be consanguineous mating. Okay? Cool? Yes, Ruhi, you're also correct. Super correct. Now, from the following pedigree chart of the family, what is this? What does this show? Now use the knowledge that you have for pedigree chart and go ahead and solve this particular question. Come on. It is autosomal dominant trait, it is autosomal recessive trait, it is autosomal dominant trait, it is allosomal, sorry, it is allosomal. Allosomal means sex-linked, okay? Allosomal means sex-linked. Sex-linked recessive trait or dominant trait. What do you think is the answer for this particular question? Come on, come on, come on. B. B is the answer. Autosome linked recessive trait. Why recessive trait? Because in every generation it is not being followed. Okay, if at all you see, there is skipping of generation. In the first generation it was not there. The second generation is there, third generation is there, but the first generation it was not there. So recessive for sure. Now, why autosomal? Because over here somehow the males and the females are equally affected in one generation. So here the female, here the male, and you do not see any crisscross pattern also. So it is why the answer for this question is going to be B. Okay, it is going to be B. Done? Yes, awesome. So how do you feel after solving some questions? So identify symbols used in the pedigree analysis. This is MQP question. You know it's consanguineous mating, and this is mating between parents producing a diseased child as well. Legend, Baba, which question are you solving? Okay. Now, over here, I have one very important question. Mendelian disorders may be autosomal or sex-linked or anything. Basically, they are saying that this is autosomal dominant disorder. And they are showing a square where the person has the disease. If the person needs to have the disease and it's an autosomal dominant disease, then what will be the genotype? Autosomal dominant. That means a dominant allele should be there. So it can be like this because the dominant allele is there, it will show the effect, or it can be like this also. Then we say that this is going to be the case. The male is being affected also, so it has to be like this. Got it? The male is being affected, and if it is being affected and it's an autosomal dominant disorder, you need a dominant allele anyway. Now, autosomal recessive. It's an autosomal recessive disorder. There's a female who is not being affected. If she is not being affected over here, that means recessive condition should not be followed because it's about an autosomal recessive disorder. If she has recessive, she will be diseased, but here she is not diseased. So again, she should be either like this or like this, where only the dominant alleles are there. Recessive alleles should not be coming together. Got it? Sex-linked dominant disorder. Again, a square is there. This is sex-linked dominant disorder, and this person is normal. If this is sex-linked dominant disorder and this person is basically normal, that means it should not have a dominant trait, rather it should be having a recessive trait, that is X small ay. If it had X capital AY, that means that person would have had the disease, but you're looking for a dominant disorder, right? And this person is not affected, that means you have to do the opposite of it. Now, sex-linked recessive disorder, and both of them are affected. If both of them are affected and it's sex-linked recessive, put the recessive character, that guy will be affected. Put the recessive characters over here, the female will also be affected. Done? Got it? It's an easy question somehow. But first time when you see, it's going to be a bit tough. So the last topic that we have is this, that is chromosomal disorders. I'm going a bit fast because I know you have another live soon at 6 o'clock. And yeah, so aneuploidy and polyploidy is the term for chromosomal disorders. So chromosomal disorders means if at all there is a loss of a chromosome, gain of a chromosome, or the structure of the chromosome is being disoriented, whatever problem it causes, it leads to chromosomal disorder. One reason is aneuploidy. Aneuploidy means failure of segregation of chromatids during cell division cycle results in loss or gain of the chromosome. The chromosomes which are there, they are not separating really well. Okay, there is no separation happening properly, there might be an extra chromosome joining up, there might be loss of a chromosome. This condition is known as aneuploidy. This happens because of failure of segregation of chromosome, failure of separation of chromosomes. On the other hand, you have polyploidy. Polyploidy means what? Failure of cytokinesis after telophase of cell division. Last phase is what? Telophase, and after that the cell division did not take place. If the cell division did not take place, what happens basically? The the chromosomes which are there, they have duplicated. You have double the amount of chromosomes over here. The cell is not dividing. That means this cell will be having double the amount of chromosome. The whole set of the chromosome is going to increase. But don't worry, this usually happens in the case of plants, not in the case of animals. Okay, so polyploidy is for plants, aneuploidy is for animals, and in aneuploidy you have trisomy condition and monosomy. Trisomy means when an additional copy of a chromosome may be included. When additional copy comes, it is trisomy. When you lose one of the chromosomes out there, that is known as monosomy. And you have some examples with which you will learn even better. So here are the example. The first one is Down syndrome. In Down syndrome, my dear children, what exactly happens? You know, the 21st chromosome, we had 23 pairs of chromosome. The 21st chromosome, which is an autosome, a normal chromosome, my dear children, you get an extra copy of it. You should have only 2, but you get an extra copy of it, which is why I'll call this as trisomy of 21st chromosome. Now over here, you should understand one very important thing. There is something that is known as karyotype, as in the numbering of the chromosome. So in general, we have 44 autosomes. 46 chromosomes are there in total, right? So 44 autosomes, and we have sex chromosomes. It can be X and Y, 45th and 46th chromosome, or it can be XX also. But in this particular condition, do you think you have 44 autosomes? No, Baba. You will have one extra autosome out there. You will be having one extra autosome, which is why you can write it something like this. 45 autosomes plus XX if it is a female who is having Down syndrome, or you can write 45 autosomes plus XY if it's a male who is having Down syndrome. And also remember, over here it's because of extra chromosome number 21. So one very important thing, if we take out a total of this, okay, if we take out a total of this, 45 chromosomes are over here, and then XY. That means 46, 47. So you can also write it like this, 47 autosomes, XX, but that won't be correct. Please write it like this. Please write it like this. I think that is more correct. So if it is not very much clear for you, I can write it properly and give it to you. It is 45A XX, or it can be XY. This is the karyotype. And over here, the features which are there, short stature will be there, round head will be there, furrowed tongue will be there, broad palm crease and mental retardation, and also delayed development. These symptoms are super important for Down syndrome or for any other chromosomal disorders out there. So it's a trisomy of 21st chromosome, 45A plus XX or XY is the karyotype out there. The features are short stature, round head, furrowed tongue means the tongue looks like almost like this, and there is mental retardation, there is broad palm crease as well. Cool, and delayed development. Now trisomy is also seen in the case of Klinefelter syndrome. Over here you need to understand, usually a male have XY chromosome, but here you will have an extra X chromosome. So trisomy is happening, but in the case of sex chromosomes or allosomes. So this is an example of allosomal trisomy. This is autosomal trisomy, this is allosomal trisomy. So over here, how do you write? 44 autosomes are there, then you have XXY condition. You can write like this, or you can make the plus thing, that is 47 XXY. In total you have 47 chromosomes, and then just put XXY. That's all. This is going to be the karyotype of Klinefelter syndrome. Remember, you have an extra X chromosome. So it was XY condition, but you had an extra X chromosome. That means basically it was supposed to be a male. So overall masculine development will be there. This is a very important question. Overall masculine development is there, but this person would be having gynecomastia, that means development of breast. That is a female character. It's what? A female character. Okay, got it? Please write, when Winter Lunar, if at all you're writing 44A, please write it as autosomes. 44A plus XXY. Or then you would have to write 47, XXY. So if you're mentioning autosomes separately, mention them separately. If you're not mentioning them, put it together, but do not mention autosomes at all. Okay? So this is overall masculine development, but with gynecomastia over here, this person is going to be sterile. They cannot produce their own babies because spermatogenesis is not going to happen. Secondary sexual characters, my dear children, are going to be incomplete. There is no proper development. Spermatogenesis is not going to happen up over here. I hope it is super clear. Whereas when you talk about Turner syndrome, things are going to be quite different. Here, instead of having XX chromosome, you lose one chromosome. There is a loss of X chromosome, that is why we call it as monosomy condition. So it will be 44 autosomes plus XO, you can write like that, or in total how many chromosomes are there? 44 and then 45. 45 XO condition. So here, it's female body itself, but this female is going to have rudimentary ovary. The ovary is not fully developed, which is why she is going to be sterile in nature. And lack of secondary sexual characteristics is also there. Don't think here you will be finding masculine features. No. Masculine features are there in the, what do you call, Klinefelter syndrome only. Got it? Done. Dusted. Clear. So these are the things, please make sure you know the karyotypes. They are super important out there, and also all the features. Now I'll be showing you a few questions. Try to solve this and then we will end this. Identify which one of the following given pair of option is correct with respect to Down syndrome and Turner's syndrome. Down syndrome and Turner syndrome. The features should be matched for both of them. For both of them. So Down syndrome, short stature, correct. Turner syndrome, gynecomastia, no, that is a symptom of Klinefelter syndrome. So that is wrong. Round head, partially open mouth, correct. Overall masculine development, again that is for Klinefelter syndrome. broad palm, physical and mental development retarded, and over here sterile females with rudimentary ovaries. This is correct for Turner syndrome. This is correct for Down syndrome also. So this has to be the correct statement. Yes, exactly. C is the answer. So over here C is there. The answer is this, Legend Soul Gaming. Awesome. Okay, going ahead, what do we have? We have one more question out there. Okay, find the incorrect statement among the following. In sex-linked recessive traits, the gene is transmitted from unaffected carrier female to some of the male progeny. In sex-linked recessive traits, the gene is transmitted from unaffected carrier female to some of the male progeny. In color blindness, you saw that it was transferred to the male progeny. This is correct, no? This is correct. In hemophilia, you also saw that carrier females are passing it. So that is also correct. Now, accumulation of phenyl pyruvic acid in the brain results in mental retardation. Yes, it does. That is also correct. Individuals affected by Down syndrome will have congenital heart defect and are more intelligent. Baba, are they more intelligent? No. In them, the mental retardation is being seen. Mental retardation is being seen. Obviously, this particular statement would be wrong to say. So this is an incorrect statement. Turner syndrome is caused due to absence of one X chromosome. Yes, that is also correct. It is because of absence of one X chromosome. So your answer for this question is C. Yes, Winter Luna. Yes, Legend Soul Gaming. Awesome. Chalo. Gynecomastia is a symptom of, this is a super easy question, Turner syndrome, Down syndrome, Cre du Chat syndrome, Klinefelter syndrome. Turner syndrome is an example of what? Gynecomastia is a symptom of what? Gynecomastia is something that you see where D, exactly, it is Klinefelter's syndrome. The answer for this question is Klinefelter's syndrome. It's Habiba, dear, I think that's a change in the question. Okay. So this is a picture given in your NCERT for gynecomastia in the case of males. Now solve this one. Match the column 1 with column 2. Autosomal trisomy, allosomal trisomy, allosomal monosomy, cystic fibrosis. What do you think is the answer for this particular question? Autosomal trisomy, allosomal monosomy, cystic fibrosis. You have to match this with this. Autosomal trisomy, the answer for that is autosome and trisomy. The answer for that will be Down syndrome. A goes with 4. It's okay, da. It's okay, Habiba. It's okay. Legend Soul Gaming says the answer for this question is D. What is D? A goes with 4. Allosomal trisomy, that means sex chromosome, one extra chromosome is there. That means Klinefelter's will be there. Allosomal monosomy, sex chromosome, one chromosome will be less. So it is going to be Turner syndrome. Cystic fibrosis, my dear children, it's a Mendelian disorder. It's a recessive disorder. Myotonic dystrophy is what? Dominant disorder. Remember that, do not be mistaken. So all of you are correct. Your answer for this question is D. Awesome, awesome, awesome. So now today I think you have studied a lot of things. Match column 1 with column 2 and find the correct answer. Aneuploidy, monoploidy, polyploidy, diploidy. Aneuploidy we know. What is aneuploidy? Loss or gain of a chromosome. A goes with 2. A goes with 2 and you just have one option with that, and that's how you find the entire answer. Monoploidy means a single set of chromosomes. This is not given in NCERT, but you need to use your understanding. Aneuploidy means this, right? Polyploidy means what? Increase in the whole set of chromosomes. This also we know. So A goes with 2 and C goes with 1. A goes with 2, C goes with 1. You just have 2 more options to match things up. So B, monoploidy will go with single set of chromosome. Diploidy will go with 2 set of chromosome. Monoploid, diploid. Okay, so your answer for this question is C. And you guys are giving me the answer. That's awesome. Okay. Now, which of the following statement is correct? Change in the whole set of chromosome is known as aneuploidy. Change in the whole set of chromosome is known as aneuploidy. Sickle cell anemia is a quantitative problem. Female carrier for hemophilia may transmit the disease to sons. Thalassaemia is a qualitative problem. Which of them is correct is being asked. Which of them? Okay, C. C is what? Female carrier for hemophilia may transmit the disease to sons. Correct. Change in the whole set of chromosome is known as polyploidy. Sickle cell anemia is a qualitative disorder, not quantitative. And thalassaemia is a quantitative disorder. So C is your answer. Yes, Winter, Luna, and also Habiba. Now, you have some model question paper questions, and that is this. Define aneuploidy, give examples for that, and mention the karyotype of the Klinefelter syndrome. I have already explained it to you. The answer is right in front of you, so you can just follow it. That would be more than enough. Okay, and also categorize the following as either autosomal or sex-linked disorders. This is given in MQP3 this year's. So hemophilia, you know, it is sex-linked. Thalassaemia, phenylketonuria, myotonic dystrophy, all of them are going to be autosomal as well. And this, what conclusion will you arrive after careful observation of the below given diagram? Valine is being substituted, is a substitution reaction. Sickle cell anemia is going to be taking place. It's point mutation. And identify the disorder by following symptom. Individual with overall muscular development, but with gynecomastia, it has to be Klinefelter's syndrome. Okay. Again, the answer is given right over here. You can watch the longer question. And this is going to be the last question of today's session. Okay, my neck is paining. A plant having the genotype capital A, small a, capital B, small b will produce dash kinds of gametes. And I have kept this question for the last because it's a new concept. It's not there. And you might, not know it as well. But if you know, you can tell me. A plant having the genotype capital A, capital B, small b will produce how many types of gametes? Surya Pratap, say, Surya Kapse, hi dear. So how many types of gametes are going to be produced? Capital A, capital B, small b. Always remember, there is a formula that is 2 raised to the power n. N stands for number of heterozygotes. If at all you see this is a heterozygote. If at all you see this is also a heterozygous condition. Capital A, capital B, small b. Both of them are heterozygous in condition. So how many of them do I have? I have n is equals to 2. So 2 raised to the power 2 is equals to what? The answer is going to be 4. So your answer for this question is going to be B. It is easy. Exactly. Cool. So, okay. That's how we end today's live. So from my end, principles of inheritance and variation is completely done. I have included everything that is super important for you. Everything that could show up in your examination. I just have one hope that you understood things. But if at all you are someone who has still some doubts left, put it down in the comment section. I am here for you. I'll explain it to you. Okay? And thank you so much for staying with me for a long time. I saw some people such as Winter, Luna, Habiba, and also Subhash were there for a very long time. Even Legend Soul Gaming. Thank you so much, guys. And you guys are doing awesome. You are hard workers. And obviously, your hard work will be paying off. Okay? Thank you so much. When is your next session, ma'am? Next session is going to be, I think, on Monday 19th. Okay? So if at all you have your friends who have not subscribed to the channel yet, please tell them. And yes, that's all. Thank you so much. I'll rest. You also please take care of yourself. And yeah, bye-bye. Do not forget to hit the subscribe button as well as the like button. Lots of love. All the best. Ta-ta.